There is no universal heritability for a characteristic. The value of heritability for a characteristic is specific for a given population in a given environment. Recall that broad sense heritability is genetic variance divided by phenotypic variance. Genetic variance depends on which genes are present, which often differs between populations. In the example of polydactyly in rabbits, there were no genes for polydactyly in the group, so the heritability of the characteristic was zero. A different group of rabbits might contain many genes for polydactyly, and the heritability of the characteristic might be high. Environmental differences may affect heritability because BP is composed of both genetic and environmental variants. Institute for Advanced Studies, Job 67. When the environmental differences that affect a characteristic differ between two groups, the heritabilities for the two groups also will often differ. Because heritability is specific to a defined population in a given environment, it is important not to extrapolate heritabilities from one population to another. For example, human height is determined by environmental factors such as nutrition and health and by genes. If we measure the heritability of height in a developed country, we might obtain a value of 0.8, indicating that the variation in height in this population is largely genetic. This population has a high heritability because most people have adequate nutrition and healthcare. B is low, so most of the phenotypic variation in height is genetically determined. It would be incorrect for us to assume that height has a high heritability in all human populations. In developing countries, there may be more variation in a range of environmental factors. Some people may enjoy good nutrition and health, whereas others may have a diet deficient in protein and suffer from diseases that affect stature. If we measure the heritability of height in such a country, we would undoubtedly obtain a lower value than we observed in the developed country because there is more environmental variation and the genetic variance in height constitutes a smaller proportion of the phenotypic variation, making the heritability lower. The important point to remember is that heritability must be calculated separately for each population and each environment. Even when heritability is high, environmental factors may influence a characteristic. High heritability does not mean that environmental factors cannot influence the expression of a characteristic. High heritability indicates only that the environmental variation to which the population is currently exposed is not responsible for variation in the characteristic. Let's look again at human height. In most developed countries, heritability of human height is high, indicating that genetic differences are responsible for most of the variation in height. It would be wrong for us to conclude that human height cannot be changed by alteration of the environment. Indeed, height decreased in several European cities during World War II owing to hunger and disease, and height can be increased dramatically by the administration of growth hormone to children. The absence of environmental variation in a characteristic does not mean that the characteristic will not respond to environmental change. Heritabilities indicate nothing about the nature of population differences in a characteristic. A common misconception about heritability is that it provides information about population differences in a characteristic. Heritability is specific for a given population in a given environment, so it cannot be used to draw conclusions about why populations differ in a characteristic. Suppose we measure heritability for human height in two groups. One group is from a small town in a developed country where everyone consumes a high protein diet. Because there is little variation in the environmental factors that affect human height and there is some genetic variation, the heritability of height in this group is high. The second group comprises the inhabitants of a single village in a developing country. The consumption of protein by these people is only 25% of that consumed by those in the first group, so their average adult height is several centimeters less than that in the developed country. Again, there is little variation in the environmental factors that determine height in this group because everyone in the village eats the same types of food and is exposed to the same diseases. Because there is little environmental variation and there is some genetic variation, the heritability of height in this group also is high. Thus, the heritability of height in both groups is high, and the average height in the two groups is considerably different. We might be tempted to conclude that the difference in height between the two groups is genetically based, that the people in the developed country are genetically taller than the people in the developing country. This conclusion is obviously wrong, however, because the differences in height are due largely to diet, an environmental factor. Heritability provides no information about the causes of differences between populations. These limitations of heritability have often been ignored, particularly in arguments about possible social implications of genetic differences between humans. Soon after Mendel's principles of heredity were rediscovered, some geneticists began to claim that many human behavioral characteristics are determined entirely by genes. This claim led to debates about whether characteristics such as human intelligence are determined by genes or environment. Many of the early claims of genetically based human behavior were based on poor research. Unfortunately, the results of these studies were often accepted at face value and led to a number of eugenic laws that discriminated against certain groups of people. Today, geneticists recognize that many behavioral characteristics are influenced by complex interaction of genes and environment and that it is very difficult to separate genetic effects from those of the environment. The results of a number of modern studies indicate that human intelligence as measured by IQ and other intelligence tests has a moderately high heritability, usually from 0.4 to 0.8. On the basis of this observation, some people have argued that intelligence is innate and that enhanced educational opportunities cannot boost intelligence. This argument is based on the misconception that, when heritability is high, changing the environment will not alter the characteristic. In addition, because heritabilities of intelligence range from 0.4 to 0.8, a considerable amount of the variance in intelligence originates from environmental differences. Another argument based on the misconception about heritability is that ethnic differences in measures of intelligence are genetically based. Because the results of some genetic studies show that IQ has moderate heritability and because other studies find differences in the average IQ of ethnic groups, some people have suggested that ethnic differences in IQ are genetically based. As in the example of the effects of diet on nutrition, heritability provides no information about causes of differences among groups. It indicates only the degree to which phenotypic variance within a single group is genetically based. High heritability for a characteristic does not mean that phenotypic differences between ethnic groups are genetic. We should also remember that separating genetic and environmental effects in humans is very difficult, so heritability estimates themselves may be unreliable. QTL mapping, locating genes that affect quantitative characteristics. The statistical methods described for use in analyzing quantitative characteristics can be used both to make predictions about the average phenotype expected in offspring and to estimate the overall contribution of genes to variation in a characteristic. These methods do not, however, allow us to identify and determine the influence of individual genes that affect quantitative characteristics. The genes that control polygenic characteristics are referred to as quantitative trait loci (QTLs). Although quantitative genetics has made important contributions to basic biology and to plant and animal breeding, the inability to identify QTLs and measure their individual effects has severely limited the application of quantitative genetic methods. Mapping QTLs in recent years, numerous genetic markers have been identified and mapped with the use of recombinant DNA techniques, making it possible to identify QTLs by linkage analysis. The underlying idea is simple. If the inheritance of a genetic marker is associated consistently with the inheritance of a particular characteristic, such as increased height, then that marker must be linked to a QTL that affects height. The key is to have enough genetic markers so that QTLs can be detected throughout the genome. With the introduction of restriction fragment length polymorphisms and microsatellite variations, variable markers are now available for mapping QTLs in a number of different organisms. A common procedure for mapping QTLs is to cross two homozygous strains that differ in alleles at many loci. The resulting F1 progeny are then intercrossed or backcrossed to allow the genes to recombine through independent assortment and crossing over. Genes on different chromosomes and genes that are far apart on the same chromosome will recombine freely. Genes that are closely linked will be inherited together. The offspring are measured for one or more quantitative characteristics. At the same time, they are genotyped for numerous genetic markers that span the genome. Any correlation between the inheritance of a particular marker allele and a quantitative phenotype indicates that a QTL is linked to that marker. If enough markers are used, it is theoretically possible to detect all the QTLs affecting a characteristic. This approach has been used to detect genes affecting various characteristics in several plant and animal species. Table 2. Applications of QTL mapping the number of genes affecting a quantitative characteristic can be estimated by locating QTLs with genetic markers and adding up the number of QTLs detected. This method will always be an underestimate because QTLs that are located close together on the same chromosome will be counted together, and those with small effects are likely to be missed. QTL mapping also provides information about the magnitude of the effects that individual genes have on a quantitative characteristic. The polygenic model assumes that many genes affect a quantitative characteristic, that the effect of each gene is small, and that the effects of the genes are equal and additive. The results of studies of QTLs in a number of organisms now show that these assumptions are not always valid. Polygens appear to vary widely in their effects. In many of the characteristics that have been studied, a few QTLs account for much of the phenotypic variation. In some instances, individual QTLs have been mapped that account for more than 20% of the variance in the characteristic. Institute for Advanced Studies, Job 69. High mutation, types, causes of detection, mutant types, lethal, conditional, biochemical, loss of function, gain of function, germinal versus somatic mutants, insertional mutagenesis. The nature of mutation. DNA is a highly stable molecule that replicates with amazing accuracy, but changes in DNA structure and errors of replication do occur. A mutation is defined as an inherited change in genetic information. The descendants may be cells produced by cell division or individual organisms produced by reproduction. The importance of mutations. Mutations are both the sustainer of life and the cause of great suffering. On the one hand, mutation is the source of all genetic variation, the raw material of evolution. Without mutations and the variation that they generate, organisms could not adapt to changing environments and would risk extinction. On the other hand, most mutations have detrimental effects, and mutation is the source of many human diseases and disorders. Much of genetics focuses on how variants produced by mutation are inherited. Genetic crosses are meaningless if all individuals are identically homozygous for the same alleles. Mutations serve as important tools of genetic analysis. The solution to almost any genetic problem begins with a good set of mutants. Much of Gregor Mendel's success in unraveling the principles of inheritance can be traced to his use of carefully selected variants of the garden pea. Similarly, Thomas Hunt Morgan and his students discovered many basic principles of genetics by analyzing mutant fruit flies. Mutations are also useful for probing fundamental biological processes. Finding mutations that affect different components of a biological system and studying their effects can often lead to an understanding of the system. This method, referred to as genetic dissection, is analogous to figuring out how an automobile works by breaking different parts of a car and observing the effects. For example, smash the radiator and the engine overheats, revealing that the radiator cools the engine. The disruption of function in individual organisms bearing particular mutations likewise can be a source of insight into biological processes. For example, geneticists have begun to unravel the molecular details of development by studying mutations that interrupt various embryonic stages in Drosophila. Although this method of breaking parts to determine a function might seem like a crude approach to understanding a system, it is actually very powerful and has been used extensively in biochemistry, developmental biology, physiology, and behavioral science, but this method is not recommended for learning how your car works. Categories of mutations. In multicellular organisms, we can distinguish between two broad categories of mutations: somatic mutations and germline mutations. Somatic mutations arise in somatic tissues, which do not produce gametes. Figure 1. These mutations are passed on to other cells through the process of mitosis, which leads to a population of genetically identical cells, a clone. The earlier in development that a somatic mutation occurs, the larger the clone of cells within an individual organism that will contain the mutation. Because of the huge number of cells present in a typical eukaryotic organism, somatic mutations must be numerous. For example, there are about 10-14 cells in the human body. If a mutation arises only once in every million cell divisions, a fairly typical rate of mutation, hundreds of millions of somatic mutations must arise in each person. The effect of these mutations depends on many factors, including the type of cell in which they occur and the developmental stage at which they arise. Many somatic mutations have no obvious effect on the phenotype of the organism because the function of the mutant cell, even the cell itself, is replaced by that of normal cells. However, cells with a somatic mutation that stimulates cell division can increase in number and spread. This type of mutation can give rise to cells with a selective advantage and is the basis for all cancers. Germline mutations arise in cells that ultimately produce gametes. These mutations can be passed to future generations, producing individual organisms that carry the mutation in all their somatic and germline cells. See figure 1. When we speak of mutations in multicellular organisms, we're usually talking about germline mutations. In single-cell organisms, however, there is no distinction between germline and somatic mutations because cell division results in new individuals. Historically, mutations have been partitioned into those that affect a single gene, called gene mutations, and those that affect the number or structure of chromosomes, called chromosome mutations. This distinction arose because chromosome mutations could be observed directly by looking at chromosomes with a microscope, whereas gene mutations could be detected only by observing their phenotypic effects. Now, with the development of DNA sequencing, gene mutations and chromosome mutations are distinguished somewhat arbitrarily on the basis of the size of the DNA lesion. Nevertheless, it is useful to use the term chromosome mutation for a large-scale genetic alteration that affects chromosome structure or the number of chromosomes, and the term gene mutation for a relatively small DNA lesion that affects a single gene. This topic focuses on gene mutations. Types of gene mutations. There are a number of ways to classify gene mutations. Some classification schemes are based on the nature of the phenotypic effect, whether the mutation alters the amino acid sequence of the protein, and if so, how. Other schemes are based on the causative agent of the mutation, and still others focus on the molecular nature of the defect. The most appropriate scheme depends on the reason for studying the mutation. Here, we will categorize mutations primarily on the basis of their molecular nature, but we will also encounter some terms that relate the causes and the phenotypic effects of mutations. Base substitutions. The simplest type of gene mutation is a base substitution, the alternation of a single nucleotide in the DNA. Figure 2a. Because of the complementary nature of the two DNA strands, when the base of one nucleotide is altered, the base of the corresponding nucleotide on the opposite strand also will be altered in the next round of replication. A base substitution therefore usually leads to a base pair substitution. Base substitutions are of two types. In a transition, a purine is replaced by a different purine, or alternatively, a pyrimidine is replaced by a different pyrimidine. Figure 3. In a transversion, a purine is replaced by a pyrimidine, or a pyrimidine is replaced by a purine. The number of possible transversions is twice the number of possible transitions, but transitions usually arise more frequently. Insertions and deletions. The second major class of gene mutations contains insertions and deletions, the addition or the removal, respectively, of one or more nucleotide pairs. Figure 2b and c. Although base substitutions are often assumed to be the most common type of mutation, molecular analysis has revealed that insertions and deletions are more frequent. Insertions and deletions within sequences that encode proteins may lead to frameshift mutations, changes in the reading frame of the gene. The initiation codon in mRNA sets the reading frame. After the initiation codon, other codons are read as successive non-overlapping groups of 3 nucleotides. The addition or deletion of a nucleotide usually changes the reading frame, altering all amino acids encoded by codons following the mutation. See figure 2b and c. Many amino acids can be affected, so frameshift mutations generally have drastic effects on the phenotype. Not all insertions and deletions lead to frameshifts. However, because codons consist of 3 nucleotides, insertions and deletions consisting of any multiple of 3 nucleotides will leave the reading frame intact, although the addition or removal of one or more amino acids may still affect the phenotype. These mutations are called in-frame insertions and deletions, respectively. Expanding nucleotide repeats in 1991, an entirely novel type of mutation was discovered. This mutation occurs in a gene called FMR1 and causes Fragile X syndrome, the most common hereditary cause of mental retardation. The disorder is so named because, in specially treated cells of persons having the condition, the tip of the X chromosome is attached only by a slender thread. The FMR1 gene contains a number of adjacent copies of the nucleotide CGG. The normal FMR1 allele not containing the mutation has 60 or fewer copies of this nucleotide, but in persons with Fragile X syndrome, the allele may harbor hundreds or even thousands of copies. Mutations in which copies of the nucleotide may increase greatly in number are called expanding nucleotide repeats. Expanding nucleotide repeats have been found in several other human diseases. Table 1. The number of copies of the nucleotide repeat often correlates with the severity or age of onset of the disease. The number of copies of the repeat also correlates with the instability of nucleotide repeats. When more repeats are present, the probability of expansion to even more repeats increases. This instability leads to a phenomenon known as intensification, in which diseases caused by nucleotide repeat expansions become more severe in each generation. Less commonly, the number of nucleotide repeats may decrease within a family. Institute for Advanced Studies, Jodhpur 71. How an increase in the number of nucleotides produces disease symptoms is not yet clear. In several of the diseases, e.g., Huntington disease, the nucleotide CAG expands within the coding part of a gene, producing a toxic protein that has extraglutamine residues, the amino acid encoded by CAG. In other diseases, e.g., Fragile X syndrome and myotonic dystrophy, the repeat is outside the coding region of the gene and therefore must have some other mode of action. At least one disease, a rare type of epilepsy, has now been associated with an expanding repeat of a 12bp sequence. Although this repeat is not a trinucleotide, it is included as a type of expanding trinucleotide because its repeat is a multiple of 3. The mechanism that leads to the expansion of trinucleotide repeats is still unclear. Strand slippage in DNA replication and crossing over between misaligned repeats are two possible sources of expansion. Single-stranded regions of some trinucleotide repeats are known to fold into hairpins, figure 4, and other special DNA structures. Such structures may promote strand slippage and replication and may prevent these errors from being recognized and corrected. Phenotypic effects of mutations. Mutations have a variety of phenotypic effects. The effect of a mutation must be considered with reference to a phenotype against which the mutant can be compared, which is usually the wild-type phenotype, that is, the most common phenotype in natural populations of the organism. For example, most Drosophila melanogaster in nature have red eyes, so red eyes are considered the wild-type eye color. Any other genetically determined eye color in fruit flies is considered to be a mutant. A mutation that alters the wild-type phenotype is called a forward mutation, whereas a reverse mutation, a reversion changes a mutant phenotype back into the wild-type. Geneticists use special terms to describe the phenotypic effects of mutations. A base substitution that alters a codon in the mRNA, resulting in a different amino acid in the protein, is referred to as a missense mutation, figure 5a. A nonsense mutation changes a sense codon, one that specifies an amino acid, into a nonsense codon, one that terminates translation, figure 5b. If a nonsense mutation occurs early in the mRNA sequence, the protein will be greatly shortened and will usually be non-functional. A silent mutation alters a codon, but thanks to the redundancy of the genetic code, the codon still specifies the same amino acid, figure 5c. A neutral mutation is a missense mutation that alters the amino acid sequence of the protein but does not change its function. Neutral mutations occur when one amino acid is replaced by another that is chemically similar or when the affected amino acid has little influence on protein function. Loss of function mutations cause a complete or partial absence of normal function. A loss of function mutation so alters the structure of the protein that the protein no longer works correctly, or the mutation can occur in regulatory regions that affect the transcription, translation, or splicing of the protein. Loss of function mutations are frequently recessive, and diploid individuals must be homozygous for the mutation before they can exhibit the effects of the loss of the functional protein. In contrast, a gain of function mutation produces an entirely new trait, or it causes the trait to appear in inappropriate tissues or at inappropriate times in development. These mutations are frequently dominant in their expression. Still other types of mutations are conditional mutations, which are expressed only under certain conditions, and lethal mutations, which cause premature death. Suppressor mutations. A suppressor mutation is a genetic change that hides or suppresses the effect of another mutation. This type of mutation is distinct from a reverse mutation, in which the mutated site changes back into the original wild type sequence, figure 6. A suppressor mutation occurs at a site that is distinct from the site of the original mutation. Thus, an individual organism with a suppressor mutation is a double mutant, possessing both the original mutation and a suppressor mutation but exhibiting the phenotype of an unmutated wild type. Geneticists distinguish between two classes of suppressor mutations, introgenic and introgenic. An introgenic suppressor is in the same gene as that containing the mutation being suppressed and may work in several ways. The suppressor may change a second nucleotide in the same codon that was altered by the original mutation, producing a codon that specifies the same amino acid as the original, unmutated codon, figure 7. Introgenic suppressors may also work by suppressing a frameshift mutation. If the original mutation is a one-base deletion, then the addition of a single base elsewhere in the gene will restore the former reading frame, see figure 7. Consider the following nucleotide sequence in DNA and the amino acids that it encodes. Suppose a one-base deletion occurs in the first nucleotide of the second codon. This deletion shifts the reading frame by one nucleotide and alters all the amino acids that follow the mutation. If a single nucleotide is added to the third codon, the suppressor mutation, the reading frame is restored, although two of the amino acids differ from those specified by the original sequence. Similarly, a mutation due to an insertion may be suppressed by subsequent deletion in the same gene. A third way in which an introgenic suppressor may work is by making compensatory changes in the protein. A first missense mutation may alter the folding of a polypeptide chain by changing the way in which amino acids in the protein interact with one another. A second missense mutation at a different site, the suppressor may recreate the original folding pattern by restoring interactions between the amino acids. Introgenic suppressors, in contrast, occur in a gene that is different from the one bearing the original mutation. These suppressors sometimes work by changing the way that the mRNA is translated. In the example illustrated in figure 8, the original DNA sequence is AAC, UUG in the mRNA. Institute for Advanced Studies, job 73. And specifies leucine. This sequence mutates to ATC, UAG in mRNA, a termination codon. The ATC nonsense mutation can be suppressed by mutation in a gene that encodes the turn molecule by changing the anticodon on the turn so that it is capable of pairing with the UAG termination codon. For example, the gene that encodes the turn for tyrosine, ternitire, which has the anticodon AUA, might be mutated to have the anticodon AUC, which will then pair with the UAG stop codon. Instead of translation terminating at the UAG codon, tyrosine would be inserted into the protein and a full-length protein would be produced, although tyrosine would now substitute for leucine. The effect of this change would depend on the role of this amino acid in the overall structure of the protein, but the effect is likely to be less detrimental than the effect of the nonsense mutation, which would halt translation prematurely. Because cells in many organisms have multiple copies of Turner genes, other unmutated copies of ternitire would remain available to recognize the tyrosine codons. However, we might expect that the terners that have undergone a suppressor mutation would also suppress the normal termination codons at the ends of coding sequences, resulting in the production of longer than normal proteins, but this event does not usually take place. Mutations in Turner genes can also suppress missense and frameshift mutations. Introgenic suppressors can also work through genic interactions. Polypeptide chains which are produced by two genes may interact to produce a functional protein. A mutation in one gene may alter the encoded polypeptide so that the interaction is destroyed and then a functional protein is no longer produced. A suppressor mutation in the second gene may produce a compensatory change in its polypeptide, therefore restoring the original interaction. Characteristics of some of the different types of mutations are summarized in table 2. Mutation rates. The frequency with which a gene changes from a wild type to a mutant is referred to as the mutation rate and is generally expressed as the number of mutations per biological unit, which may be mutations per cell division, per gamete, or per round of replication. For example, the mutation rate for achondroplasia, a type of hereditary dwarfism, is about 4 mutations per 100,000 gametes, usually expressed more simply as 4 x 10^5. In contrast, mutation frequency is defined as the incidence of a specific type of mutation within a group of individual organisms. For achondroplasia, the mutation frequency in the United States is about 2 x 10^4, which means that about 1 of every 20,000 persons in the US population carries this mutation. Mutation rates are affected by 3 factors. First, they depend on the frequency with which primary changes take place in DNA. Primary change may arise from spontaneous molecular changes in DNA, or it may be induced by chemical or physical agents in the environment. A second factor influencing the mutation rate is the probability that, when a change takes place, it will be repaired. Most cells possess a number of mechanisms to repair altered DNA, so most alterations are corrected before they are replicated. If these repair systems are effective, mutation rates will be low. If they are faulty, mutation rates will be elevated. Some mutations increase the overall rate of mutation in other genes. These mutations usually occur in genes that encode components of the replication machinery or DNA repair enzymes. A third factor, one that influences our ability to calculate mutation rates, is the probability that a mutation will be recognized and recorded. When DNA is sequenced, all mutations are potentially detectable. In practice, however, sequencing is expensive, so mutations are usually detected by their phenotypic effects. Some mutations may appear to arise at a higher rate simply because they are easier to detect. Mutation rates vary among organisms and among genes within organisms, table 3, but we can draw several general conclusions about mutation rates. First, spontaneous mutation rates are low for all organisms studied. Typical mutation rates for viral and bacterial genes range from about 1 to 100 mutations per 10 billion cells, 1 x 10^8 to 1 x 10^10. The mutation rates for most eukaryotic genes are a bit higher, from about 1 to 10 mutations per million gametes, 1 x 10^5 to 1 x 10^6. These higher values in eukaryotes may be due to the fact that the rates are calculated per gamete, and several cell divisions are required to produce a gamete, whereas mutation rates in prokaryotic cells and viruses are calculated per cell division. Within each major class of organisms, mutation rates vary considerably. These differences may be due to different abilities to repair mutations, unequal exposures to mutagens, or biological differences in rates of spontaneously arising mutations. Even within a single species, spontaneous rates of mutation vary among genes. The reason for this variation is not entirely understood, but some regions of DNA are known to be more susceptible to mutation than others. Causes of mutations. Mutations result from both internal and external factors. Those that are a result of natural changes in DNA structure are termed spontaneous mutations, whereas those that result from changes caused by environmental chemicals or radiation are induced mutations. Spontaneous replication errors. Replication is amazingly accurate. Fewer than 1 in a billion errors are made in the course of DNA synthesis. However, spontaneous replication errors do occasionally occur. The primary cause of spontaneous replication errors was formerly thought to be tautomeric shifts, in which the positions of protons in the DNA bases change. Purine and pyrimidine bases exist in different chemical forms called tautomers, figure 9a. The two tautomeric forms of each base are in dynamic equilibrium, although one form is more common than the other. The standard Watson and Crick base pairings, adenine with thymine and cytosine with guanine, are between the common forms of the bases, but if the bases are in their rare tautomeric forms, other base pairings are possible, figure 9b. Watson and Crick proposed that tautomeric shifts might produce mutations, and for many years their proposal was the accepted model for spontaneous replication errors, but there has never been convincing evidence that the rare tautomers are the cause of spontaneous mutations. Furthermore, research now shows little evidence of these structures in DNA. These structures have been detected in DNA molecules and are now thought to be responsible for many of the mispairings in replication. When a mismatched base has been incorporated into a newly synthesized nucleotide chain, an incorporated error is said to have occurred. Suppose that, in replication, thymine, which normally pairs with adenine, mispairs with guanine through wobble, figure 11. In the next round of replication, the two mismatched bases separate, and each serves as template for the synthesis of a new nucleotide strand. This time, thymine pairs with adenine, producing another copy of the original DNA sequence. On the other strand, however, the incorrectly incorporated guanine serves as a template and pairs with cytosine, producing a new DNA molecule that has an incorporated error, a CG pair in place of the original T pair, a TACG base substitution. The original incorporated error leads to a replication error, which creates a permanent mutation, because all the base pairings are correct and there is no mechanism for repair systems to detect the error. Mutations due to small insertions and deletions also may arise spontaneously in replication and crossing over. Strand slippage may occur when one nucleotide strand forms a small loop, figure 12. Mispairing can also occur through wobble, in which normal, protonated, and other forms of the bases are able to pair because of flexibility in the DNA helical structure, figure 10. If the looped out nucleotides are on the newly synthesized strand, an insertion results. At the next round of replication, the insertion will be incorporated into both strands of the DNA molecule. If the looped out nucleotides are on the template strand, then there is a deletion on the newly replicated strand, and this deletion will be perpetuated in subsequent rounds of replication during normal crossing over the homologous sequences of the two DNA molecules. Align and crossing over produces no net change in the number of nucleotides in either molecule. Misaligned pairing may cause unequal crossing over, which results in one DNA molecule with an insertion and the other with a deletion. Figure 13. Institute for Advanced Studies, Jodhpur 75. Some DNA sequences are more likely than others to undergo strand slippage or unequal crossing over. Stretches of repeated sequences, such as nucleotide repeats or homopolymeric repeats, more than 5 repeats of the same base in a row are prone to strand slippage. Stretches with more repeats are more likely to undergo strand slippage. Duplicated or repetitive sequences may misalign during pairing, leading to unequal crossing over. Both strand slippage and unequal crossing over produce duplicated copies of sequences, which in turn promote further strand slippage and unequal crossing over. This chain of events may explain the phenomenon of anticipation often observed for expanding through nucleotide repeats. Spontaneous chemical changes. In addition to spontaneous mutations that arise in replication, mutations also result from spontaneous chemical changes in DNA. One such change is depurination, the loss of a purine base from a nucleotide. Depurination results when a covalent bond connecting the purine to the 1' carbon atom of the deoxyribose sugar breaks. Figure 14a producing an epurinic site, a nucleotide that lacks its purine base. An epurinic site cannot act as a template for a complementary base in replication. In the absence of base pairing constraints, an incorrect nucleotide, most often adenine, is incorporated into the newly synthesized DNA strand opposite the epurinic site. Figure 14b frequently leading to an incorporated error. The incorporated error is then transformed into a replication error at the next round of replication. Depurination is a common cause of spontaneous mutation. A mammalian cell in culture loses approximately 10,000 purines every day. Another spontaneously occurring chemical change that takes place in DNA is deamination, the loss of an amino group NH2 from a base. Deamination may occur spontaneously or be induced by mutagenic chemicals. Deamination may alter the pairing properties of a base. The deamination of cytosine, for example, produces uracil. Figure 15a, which pairs with adenine during replication. After another round of replication, the adenine will pair with thymine, creating a TA pair in place of the original CG pair. Circuit GUATA. This chemical change is a transition mutation. This type of mutation is usually repaired by enzymes that remove uracil whenever it is found in DNA. The ability to recognize the product of cytosine deamination may explain why thymine, not uracil, is found in DNA. Some cytosine bases in DNA are naturally methylated and exist in the form of 5-methylcytosine, 5 millicoulombs, which when deaminated becomes thymine. Figure 15b. Because thymine pairs with adenine in replication, the deamination of 5-methylcytosine changes an original CG pair to TA. Circuit G 5 millicoulombs, ATA. This change cannot be detected by DNA repair systems because it produces a normal base. Consequently, CGT, A transitions occur frequently in eukaryotic cells. Institute for Advanced Studies, Jodhpur 77. Chemically induced mutations. Although many mutations arise spontaneously, a number of environmental agents are capable of damaging DNA, including certain chemicals and radiation. Any environmental agent that significantly increases the rate of mutation above the spontaneous rate is called a mutagen. The first discovery of a chemical mutagen was made by Charlotte Auerbach, who was born in Germany to a Jewish family in 1899. After attending university in Berlin and doing research, she spent several years teaching at various schools in Berlin. Faced with increasing antisemitism in Nazi Germany, Auerbach immigrated to Britain, where she conducted research on the development of mutants in Drosophila. There she met Hermann Muller, who had shown that radiation induces mutations. He suggested that Auerbach try to obtain mutants by treating Drosophila with chemicals. Her initial attempts met with little success. Other scientists were conducting top secret research on mustard gas, used as a chemical weapon in World War I, and noticed that it produced many of the same effects as radiation. Auerbach was asked to determine whether mustard gas was mutagenic. Collaborating with pharmacologist J. M. Robson, Auerbach studied the effects of mustard gas on Drosophila melanogaster. The experimental conditions were crude. They heated liquid mustard gas over a Bunsen burner on the roof of the pharmacology building, and the flies were exposed to the gas in a large chamber. After developing serious burns on her hands from the gas, Auerbach let others carry out the exposures, and she analyzed the flies. Auerbach and Robson showed that mustard gas is indeed a powerful mutagen, reducing the viability of gametes and increasing the numbers of mutations seen in the offspring of exposed flies. Because the research was part of the secret war effort, publication of their findings was delayed until 1947. Base analogs. One class of chemical mutagens consists of base analogs, chemicals with structure similar to that of any of the 4 standard bases of DNA. DNA polymerases cannot distinguish these analogs from the standard bases, so if base analogs are present during replication, they may be incorporated into newly synthesized DNA molecules. For example, 5-bromoaryl 5-BU is an analog of thymine. It has the same structure as that of thymine except that it has a bromine bridge atom on the 5-carbon atom instead of a methyl group. Figure 16a. Normally, 5-bromoaryl pairs with adenine just as thymine does, but it occasionally mispairs with guanine. Figure 16b, leading to a transition TA5BU, A5BU, GCG as shown in figure 17. Through mispairing, 5-bromoaryl may also be incorporated into a newly synthesized DNA strand opposite guanine. In the next round of replication, 5-bromoaryl may pair with adenine, leading to another transition GCG5BU A.5BU AT. Another mutagenic chemical is 2-aminopurine 2AP, which is the base analog of adenine. Figure 18. Normally, 2-aminopurine base pairs with thymine, but it may mispair with cytosine, causing a transition mutation TAT.2APC.2APCG. Alternatively, 2-aminopurine may be incorporated through mispairing into the newly synthesized DNA opposite cytosine and later pair with thymine, leading to a CGC.2APT.2APT, A transition. Thus, both 5-bromoaryl and 2-aminopurine can produce transition mutations. In the laboratory, mutations by base analogs can be reversed by treatment with the same analog or by treatment with a different analog. Alkylating agents. Alkylating agents are chemicals that donate alkyl groups. These agents include methyl CH3 and ethyl CH3CH2 groups, which are added to nucleotide bases by some chemicals. For example, ethylmethane sulfonate EMS adds an ethyl group to guanine, producing 6-ethylguanine, which pairs with thymine. Thus, EMS produces CGT, A transitions. EMS is also capable of adding an ethyl group to thymine, producing 4-ethylthymine, which then pairs with guanine, leading to a TACG transition. Because EMS produces both CGTA and TACG transitions, mutations produced by EMS can be reversed by additional treatment with EMS. Mustard gas is another alkylating agent. Deamination. In addition to its spontaneous occurrence, see figure 15, deamination can be induced by some chemicals. For instance, nitrous acid deaminates cytosine, creating uracil, which in the next round of replication pairs with adenine. See figure 18b, producing a CGT, A transition mutation. Nitrous acid changes adenine into hypoxanthine, which pairs with cytosine, leading to a TACG transition. Nitrous acid also deaminates guanine, producing xanthine, which pairs with cytosine just as guanine does. However, xanthine may also pair with thymine, leading to a CGT, A transition. Nitrous acid produces exclusively transition mutations and, because both CGTA and TACG transitions are produced, these mutations can be reversed with nitrous acid. Hydroxylamine. Hydroxylamine is a very specific base-modifying mutagen that adds a hydroxyl group to cytosine, converting it into hydroxylaminocytosine. See figure 18c. This conversion increases the frequency of a rare tautomer that pairs with adenine instead of guanine and leads to CGT, A transitions. Because hydroxylamine acts only on cytosine, it will not generate TACG transitions. Thus, hydroxylamine will not reverse the mutations that it produces. Oxidative reactions. Reactive forms of oxygen, including superoxide radicals, hydrogen peroxide, and hydroxyl radicals, are produced in the course of normal aerobic metabolism, as well as by radiation, ozone, peroxides, and certain drugs. These reactive forms of oxygen damage DNA and induce mutations by bringing about chemical changes to DNA. Within each major class of organisms, mutation rates vary considerably. These differences may be due to different abilities to repair mutations, unequal exposures to mutagens, or biological differences in rates of spontaneously arising mutations. Even within a single species, spontaneous rates of mutation vary among genes. The reason for this variation is not entirely understood, but some regions of DNA are known to be more susceptible to mutation than others. Causes of mutations. Mutations result from both internal and external factors. Those that are a result of natural changes in DNA structure are termed spontaneous mutations, whereas those that result from changes caused by environmental chemicals or radiation are induced mutations. Spontaneous replication errors. Replication is amazingly accurate. Fewer than 1 in a billion errors are made in the course of DNA synthesis. However, spontaneous replication errors do occasionally occur. The primary cause of spontaneous replication errors was formerly thought to be tautomeric shifts, in which the positions of protons in the DNA bases change. Purine and pyrimidine bases exist in different chemical forms called tautomers, figure 9a. The two tautomeric forms of each base are in dynamic equilibrium, although one form is more common than the other. The standard Watson and Crick base pairings, adenine with thymine and cytosine with guanine, are between the common forms of the bases, but if the bases are in the rare tautomeric forms, other base pairings are possible, figure 9b. Watson and Crick proposed that tautomeric shifts might produce mutations, and for many years the proposal was the accepted model for spontaneous replication errors, but there has never been convincing evidence that the rare tautomers are the cause of spontaneous mutations. Furthermore, research now shows little evidence of these structures in DNA. These structures have been detected in DNA molecules and are now thought to be responsible for many of the mispairings in replication. When a mismatched base has been incorporated into a newly synthesized nucleotide chain, an incorporated error is said to have occurred. Suppose that, in replication, thymine, which normally pairs with adenine, mispairs with guanine through wobble, figure 11. In the next round of replication, the two mismatched bases separate, and each serves as template for the synthesis of a new nucleotide strand. This time, thymine pairs with adenine, producing another copy of the original DNA sequence. On the other strand, however, the incorrectly incorporated guanine serves as a template and pairs with cytosine, producing a new DNA molecule that has an incorporated error, a CG pair in place of the original TF pair, a TACG base substitution. The original incorporated error leads to a replication error, which creates a permanent mutation, because all the base pairings are correct and there is no mechanism for repair systems to detect the error. Mutations due to small insertions and deletions also may arise spontaneously in replication and crossing over. Strand slippage may occur when one nucleotide strand forms a small loop, figure 12. Mispairing can also occur through wobble, in which normal, protonated, and other forms of the bases are able to pair because of flexibility in the DNA helical structure, figure 10. If the looped out nucleotides are on the newly synthesized strand, an insertion results. At the next round of replication, the insertion will be incorporated into both strands of the DNA molecule. If the looped out nucleotides are on the template strand, then there is a deletion on the newly replicated strand, and this deletion will be perpetuated in subsequent rounds of replication. During normal crossing over, the homologous sequences of the two DNA molecules align, and crossing over produces no net change in the number of nucleotides in either molecule. Misaligned pairing may cause unequal crossing over, which results in one DNA molecule with an insertion and the other with a deletion. Figure 13. Institute for Advanced Studies, job for 75. Some DNA sequences are more likely than others to undergo strand slippage or unequal crossing over. Stretches of repeated sequences, such as for nucleotide repeats or homopolymeric repeats, more than 5 repeats of the same base in a row are prone to strand slippage. Stretches with more repeats are more likely to undergo strand slippage. Duplicated or repetitive sequences may misalign during pairing, leading to unequal crossing over. Both strand slippage and unequal crossing over produce duplicated copies of sequences, which in turn promote further strand slippage and unequal crossing over. This chain of events may explain the phenomenon of anticipation often observed for expanding for nucleotide repeats. Spontaneous chemical changes. In addition to spontaneous mutations that arise in replication, mutations also result from spontaneous chemical changes in DNA. One such change is depurination, the loss of a purine base from a nucleotide. Depurination results when a covalent bond connecting the purine to the 1' carbon atom of the deoxyribose sugar breaks, figure 14a, producing an apurinic site, a nucleotide that lacks its purine base. An apurinic site cannot act as a template for a complementary base in replication. In the absence of base pairing constraints, an incorrect nucleotide, most often adenine, is incorporated into the newly synthesized DNA strand opposite the apurinic site, figure 14b, frequently leading to an incorporated error. The incorporated error is then transformed into a replication error at the next round of replication. Depurination is a common cause of spontaneous mutation. A mammalian cell in culture loses approximately 10,000 purines every day. Another spontaneously occurring chemical change that takes place in DNA is deamination, the loss of an amino group NH2 from a base. Deamination may occur spontaneously or be induced by mutagenic chemicals. Deamination may alter the pairing properties of a base. The deamination of cytosine, for example, produces uracil, figure 15a, which pairs with adenine during replication. After another round of replication, the adenine will pair with thymine, creating a TF pair in place of the original CG pair, circuit GUATA. This chemical change is a transition mutation. This type of mutation is usually repaired by enzymes that remove uracil whenever it is found in DNA. The ability to recognize the product of cytosine deamination may explain why thymine, not uracil, is found in DNA. Some cytosine bases in DNA are naturally methylated and exist in the form of 5-methylcytosine, 5 millicoulombs, which when deaminated becomes thymine, figure 15b. Because thymine pairs with adenine in replication, the deamination of 5-methylcytosine changes an original CG pair to TA, circuit G 5 millicoulombs, ATA. This change cannot be detected by DNA repair systems because it produces a normal base. Consequently, CGT, A transitions occur frequently in eukaryotic cells. Institute for Advanced Studies, job for 77. Chemically induced mutations. Although many mutations arise spontaneously, a number of environmental agents are capable of damaging DNA, including certain chemicals and radiation. Any environmental agent that significantly increases the rate of mutation above the spontaneous rate is called a mutagen. The first discovery of a chemical mutagen was made by Charlotte Auerbach, who was born in Germany to a Jewish family in 1899. After attending university in Berlin and doing research, she spent several years teaching at various schools in Berlin. Faced with increasing anti-Semitism in Nazi Germany, Auerbach immigrated to Britain, where she conducted research on the development of mutants in Drosophila. There she met Hermann Muller, who had shown that radiation induces mutations. He suggested that Auerbach try to obtain mutants by treating Drosophila with chemicals. Her initial attempts met with little success. Other scientists were conducting top-secret research on mustard gas, used as a chemical weapon in World War I, and noticed that it produced many of the same effects as radiation. Auerbach was asked to determine whether mustard gas was mutagenic. Collaborating with pharmacologist J. M. Robson, Auerbach studied the effects of mustard gas on Drosophila melanogaster. The experimental conditions were crude. They heated liquid mustard gas over a Bunsen burner on the roof of the pharmacology building, and the flies were exposed to the gas in a large chamber. After developing serious burns on her hands from the gas, Auerbach let others carry out the exposures, and she analyzed the flies. Auerbach and Robson showed that mustard gas is indeed a powerful mutagen, reducing the viability of gametes and increasing the numbers of mutations seen in the offspring of exposed flies. Because the research was part of the secret war effort, publication of their findings was delayed until 1947. Base analogs. One class of chemical mutagens consists of base analogs, chemicals with structures similar to that of any of the 4 standard bases of DNA. DNA polymerases cannot distinguish these analogs from the standard bases, so if base analogs are present during replication, they may be incorporated into newly synthesized DNA molecules. For example, 5-bromoaryl 5-BU is an analog of thymine. It has the same structure as that of thymine except that it has a bromine bridge atom on the 5 carbon atom instead of a methyl group, figure 16a. Normally, 5-bromoaryl pairs with adenine just as thymine does, but it occasionally mispairs with guanine, figure 16b, leading to a transition TA5BU, A5BU, GCG as shown in figure 17. Through mispairing, 5-bromoaryl may also be incorporated into a newly synthesized DNA strand opposite guanine. In the next round of replication, 5-bromoaryl may pair with adenine, leading to another transition GCG5BU 8.5BU AT. Another mutagenic chemical is 2-aminopurine 2AP, which is a base analog of adenine, figure 18. Normally, 2-aminopurine base pairs with thymine, but it may mispair with cytosine, causing a transition mutation TAT.2APC.2APCG. Alternatively, 2-aminopurine may be incorporated through mispairing into the newly synthesized DNA opposite cytosine and later pair with thymine, leading to a CGC.2APT.2APT, A transition. Thus, both 5-bromoaryl and 2-aminopurine can produce transition mutations. In the laboratory, mutations by base analogs can be reversed by treatment with the same analog or by treatment with a different analog. Alkylating agents. Alkylating agents are chemicals that donate alkyl groups. These agents include methyl CH3 and ethyl CH3CH2 groups, which are added to nucleotide bases by some chemicals. For example, ethylmethane sulfonate EMS adds an ethyl group to guanine, producing 6-ethylguanine, which pairs with thymine. Thus, EMS produces CGT, 8 transitions. EMS is also capable of adding an ethyl group to thymine, producing 4-ethylthymine, which then pairs with guanine, leading to a TACG transition. Because EMS produces both CGTA and TACG transitions, mutations produced by EMS can be reversed by additional treatment with EMS. Mustard gas is another alkylating agent. Deamination. In addition to its spontaneous occurrence, see figure 15, deamination can be induced by some chemicals. For instance, nitrous acid deaminates cytosine, creating uracil, which in the next round of replication pairs with adenine, see figure 18b, producing a CGT, a transition mutation. Nitrous acid changes adenine into ipoxanthine, which pairs with cytosine, leading to a TACG transition. Nitrous acid also deaminates guanine, producing xanthine, which pairs with cytosine just as guanine does. However, xanthine may also pair with thymine, leading to a CGT, a transition. Nitrous acid produces exclusively transition mutations, and because both CGTA and TACG transitions are produced, these mutations can be reversed with nitrous acid. Hydroxylamine. Hydroxylamine is a very specific base-modifying mutagen that adds a hydroxyl group to cytosine, converting it into hydroxylaminocytosine. See figure 18c. This conversion increases the frequency of a rare trimer that pairs with adenine instead of guanine and leads to CGT, 8 transitions. Because hydroxylamine acts only on cytosine, it will not generate TACG transitions. Thus, hydroxylamine will not reverse the mutations that it produces. Oxidative reactions. Reactive forms of oxygen, including superoxide radicals, hydrogen peroxide, and hydroxyl radicals, are produced in the course of normal aerobic metabolism, as well as by radiation, ozone, peroxides, and certain drugs. These reactive forms of oxygen damage DNA and induce mutations by bringing about chemical changes to DNA. For example, oxidation converts guanine into 8-OXY7, 8-dihydrodeoxyguanine, figure 19, which frequently mispairs with adenine instead of cytosine, causing a GCT, 8 transversion mutation. Intercalating agents, such as proplavin, acridine orange, actinium bromide, and dioxin are about the same size as the nucleotide, figure 20a. They produce mutations by sandwiching themselves, intercalating between adjacent bases in DNA, distorting the three-dimensional structure of the helix and causing single nucleotide insertions and deletions in replication, figure 20b. These insertions and deletions frequently produce frameshift mutations, which change all amino acids downstream of the mutation, and so the mutagenic effects of intercalating agents are often severe. Because intercalating agents generate both additions and deletions, they can reverse the effects of their own mutations. Radiation. In 1927, Hermann Miller demonstrated that mutations in fruit flies could be induced by X-rays. The results of subsequent studies showed that X-rays greatly increase mutation rates in all organisms. The high energies of X-rays, gamma rays, and cosmic rays are all capable of penetrating tissues and damaging DNA. These forms of radiation, called ionizing radiation, dislodge electrons from the atoms that they encounter, changing stable molecules into free radicals and reactive ions, which then alter the structures of bases and break phosphodiester bonds in DNA. Ionizing radiation also frequently results in double-strand breaks in DNA. Attempts to repair these breaks can produce chromosome mutations. Ultraviolet light has less energy than that of ionizing radiation and does not eject electrons and cause ionization, but is nevertheless highly mutagenic. Purine and pyrimidine bases readily absorb UV light, resulting in the formation of chemical bonds between adjacent pyrimidine molecules on the same strand of DNA and in the creation of structures called pyrimidine dimers, figure 22a. Pyrimidine dimers consisting of two thymine bases called thymine dimers are most frequent, but cytosine dimers and thymine-cytosine dimers also can form. These dimers distort the configuration of DNA, figure 22b, and often block replication. Most pyrimidine dimers are immediately repaired by mechanisms, but some escape repair and inhibit replication and transcription. Institute for Advanced Studies, job number 79. When pyrimidine dimers block replication, cell division is inhibited and the cell usually dies. For this reason, UV light kills bacteria and is an effective sterilizing agent. For a mutation, a hereditary error in the genetic instructions, to occur, the replication block must be overcome. How do bacteria and other organisms replicate despite the presence of thymine dimers? Bacteria can circumvent replication blocks produced by pyrimidine dimers and other types of DNA damage by means of the SOS system. This system allows replication blocks to be overcome, but in the process makes numerous mistakes and greatly increases the rate of mutation. Indeed, the very reason that replication can proceed in the presence of a block is that the enzymes in the SOS system do not strictly adhere to the base pairing rules. The trade-off is that replication may continue and the cell survives, but only by sacrificing the normal accuracy of DNA synthesis. The SOS system is complex, including the products of at least 25 genes. A protein called RECA binds to the damaged DNA of the blocked replication fork and becomes activated. This activation promotes the binding of a protein called LEXA, which is a repressor of the SOS system. The activated RECA complex induces LEXA to undergo self-cleavage, destroying its repressive activity. This inactivation enables other SOS genes to be expressed, and the products of these genes allow replication of the damaged DNA to proceed. The SOS system allows bases to be inserted into a new DNA strand in the absence of bases on the template strand, but these insertions result in numerous errors in the base sequence. Eukaryotic cells have a specialized DNA polymerase called polymerase A that bypasses pyrimidine dimers. Polymerase A preferentially inserts opposite of pyrimidine dimer. This strategy seems to be reasonable because about 2/3 of pyrimidine dimers are thymine dimers. However, the insertion of AA opposite of CT dimer results in a CG-AT transversion. Polymerase A is therefore said to be an error-prone polymerase. The study of mutations. Because mutations often have detrimental effects, they have been the subject of intense study by geneticists. These studies have included the analysis of reverse mutations, which are often sources of important insight into how mutations cause DNA damage, the development of tests to determine the mutagenic properties of chemical compounds, and the investigation of human populations tragically exposed to high levels of radiation. The analysis of reverse mutations. The study of reverse mutations, reversions, can provide useful information about how mutagens alter DNA structure. For example, any mutagen that produces both ATGC and GC-A-T transitions should be able to reverse its own mutations. However, if the mutagen produces only GC-AT transitions, then reversion by the same mutagen is not possible. Hydroxylamine, see figure 18c, exhibits this type of one-way mutagenic activity. It causes GC-AT transitions but is incapable of reversing the mutations that it produces, so we know that it does not produce ATGC transitions. Ethylmethane sulfonate, see figure 18a, on the other hand, produces GC-AT transitions and reverses its own mutations, so we know that it also produces TACG transitions. Analyses of the ability of different mutagens to cause reverse mutations can be sources of insight into the molecular nature of the mutations. We can use reverse mutations to determine whether a mutation results from a base substitution or a frameshift. Base analogs such as 2-aminopurine cause transitions, and intercalating agents such as acridine orange, see figure 20, produce frameshifts. If a chemical reverses mutations produced by 2-aminopurine but not those produced by acridine orange, we can conclude that the chemical causes transitions and not frameshifts. If nitrous acid, which produces both GC-AT and ATGC transitions, reverses mutations produced by the chemical hydroxylamine, which causes only GC-AT transitions, does not, we know that, like hydroxylamine, the chemical produces only GC-AT transitions. Table 4 illustrates the reverse mutations that are theoretically possible among several mutagenic agents. The actual ability of mutagens to produce reversals is more complex than suggested by Table 4 and depends on environmental conditions and the organism tested. Detecting mutations with the Ames test. Humans and industrial societies are surrounded by a multitude of artificially produced chemicals. More than 50,000 different chemicals are in commercial and industrial use today, and from 500 to 1,000 new chemicals are introduced each year. Some of these chemicals are potential carcinogens and may cause potential harm to humans. How can we determine which chemicals are hazardous? In a few instances, previous human exposure to a specific chemical is correlated with an increase in cancer incidence, providing good evidence that the chemical is a carcinogen. But, ideally, we would like to know which chemicals are hazardous before we are exposed to them. One method for testing the cancer-causing potential of chemicals is to administer them to laboratory animals, rats or mice, and compare the incidence of cancer in the treated animals with that of control animals. These tests are unfortunately time-consuming and expensive. Furthermore, the ability of a substance to cause cancer in rodents is not always indicative of its effect on humans. After all, we aren't rats. In 1974, Bruce Ames developed a simple test for evaluating the potential of chemicals to cause cancer. The Ames test is based on the principle that both cancer and mutations result from damage to DNA, and the results of experiments have demonstrated that 90% of known carcinogens are also mutagens. Ames proposed that mutagenesis in bacteria could serve as an indicator of carcinogenesis in humans. The Ames test uses 4 strains of the bacterium Salmonella type Murium that have defects in the lipopolysaccharide coat, which normally protects the bacteria from chemicals in the environment. Furthermore, their DNA repair system has been inactivated, enhancing their susceptibility to mutagens. In 1974, Bruce Ames developed a simple test for evaluating the potential of chemicals to cause cancer. The Ames test is based on the principle that both cancer and mutations result from damage to DNA, and the results of experiments have demonstrated that 90% of known carcinogens are also mutagens. Ames proposed that mutagenesis in bacteria could serve as an indicator of carcinogenesis in humans. The Ames test uses 4 strains of the bacterium Salmonella type Murium that have defects in the lipopolysaccharide coat, which normally protects the bacteria from chemicals in the environment. Furthermore, their DNA repair system has been inactivated, enhancing their susceptibility to mutagens. One of the 4 strains used in the Ames test detects base pair substitutions. The other 3 detect different types of frameshift mutations. Each strain carries a mutation that renders it unable to synthesize the amino acid histidine, his, and the bacteria are plated onto medium that lacks histidine, figure 23. Only bacteria that have undergone a reverse mutation of the histidine gene, his plus, are able to synthesize histidine and grow on the medium. Different dilutions of a chemical to be tested are added to plates inoculated with the bacteria, and the number of mutant bacterial colonies that appear on each plate is compared with the number that appear on control plates with no chemical arose through spontaneous mutation. Any chemical that significantly increases the number of colonies appearing on a treated plate is mutagenic and is probably also carcinogenic. Some compounds are not active carcinogens but may be converted into cancer-causing compounds in the body. To make the Ames test sensitive for such potential carcinogens, a compound to be tested is first incubated in a mammalian liver extract that contains metabolic enzymes. The Ames test has been applied to thousands of chemicals in commercial products. An early demonstration of its usefulness was the discovery, in 1975, that most hair dyes sold in the United States contain compounds that were mutagenic to bacteria. These compounds were then removed from most hair dyes. Institute for Advanced Studies, Job 81. J. Structural and numerical alterations of chromosomes, deletion, duplication, inversion, translocation, ploidy and their genetic implications. Chromosome variation. Before we consider the different types of chromosome mutations, their effects, and how they arise, we will review the basics of chromosome structure. Chromosome morphology. Each functional chromosome has a centromere, where spindle fibers attach, and two telomeres that stabilize the chromosome. Chromosomes are classified into four basic types: metacentric, in which the centromere is located approximately in the middle, and so the chromosome has two arms of equal length; submetacentric, in which the centromere is displaced toward one end, creating a long arm and a short arm; acrocentric, in which the centromere is near one end, producing a long arm and a knob, or satellite, at the other; and telecentric, in which the centromeres are very near the end of the chromosome. On human chromosomes, the short arm is designated by the letter P and the long arm by the letter Q. The complete set of chromosomes that an organism possesses is called its karyotype and is usually presented as a picture of metaphase chromosomes lined up in descending order of their size, figure 1. Karyotypes are prepared from actively dividing cells, such as white blood cells, bone marrow cells, or cells from meristematic tissues of plants. After treatment with a chemical such as colchicine that prevents them from entering anaphase, the cells are chemically preserved, spread on a microscope slide, stained, and photographed. The photograph is then enlarged, and the individual chromosomes are cut out and arranged in a karyotype. For human chromosomes, karyotypes are often routinely prepared by automated machines, which scan a slide with a video camera attached to a microscope, looking for chromosome spreads. When a spread has been located, the camera takes a picture of the chromosomes, the image is digitized, and the chromosomes are sorted and arranged electronically by a computer. Preparation and staining techniques have been developed to help distinguish among chromosomes of similar size and shape. For instance, chromosomes may be treated with enzymes that partly digest them, staining with a special dye called G bands, which distinguish areas of DNA that are rich in adenine thymine base pairs, figure 2a. Q bands, figure 2b, are revealed by staining chromosomes with quinacridone mustard and viewing the chromosomes under UV light. Other techniques reveal C bands, figure 2c, which are regions of DNA occupied by centromeric heterochromatin, and R bands, figure 2b, which are rich in guanine, cytosine base pairs. Types of chromosome mutations. Chromosome mutations can be grouped into three basic categories: chromosome rearrangements, aneuploids, and polyploids. Chromosome rearrangements alter the structure of chromosomes. For example, a piece of a chromosome might be duplicated, deleted, or inverted. In aneuploidy, the number of chromosomes is altered. One or more individual chromosomes are added or deleted. In polyploidy, one or more complete sets of chromosomes are added. Some organisms, such as yeast, possess a single chromosome set, 1n, for most of their life cycles and are referred to as haploid, whereas others possess two chromosome sets and are referred to as diploid, 2n. A polyploid is any organism that has more than two sets of chromosomes, 3n, 4n, 5n, or more. Chromosome rearrangements. Chromosome rearrangements are mutations that change the structure of individual chromosomes. The four basic types of rearrangements are duplications, deletions, inversions, and translocations, figure 3. Duplications. A chromosome duplication is a mutation in which part of the chromosome has been doubled, see figure 3a. Consider a chromosome with segments A, B, C, D, E, F, G, in which represents the centromere. A duplication might include the EF segments, giving rise to a chromosome with segments A, B, C, D, F, G. This type of duplication, in which the duplicated region is immediately adjacent to the original segment, is called a tandem duplication. If the duplicated segment is located some distance from the original segment, either on the same chromosome or on a different one, this type is called a displaced duplication. An example of a displaced duplication would be A, B, C, D, F. A duplication can either be in the same orientation as the original sequence, as in the two preceding examples, or be inverted, A, B, C, D, F. When the duplication is inverted, it is called a reverse duplication. An individual homozygous for rearrangement carries the rearrangement, the mutated sequence, on both homologous chromosomes, and an individual heterozygous for rearrangement has one unmutated chromosome and one chromosome with a rearrangement. In the heterozygotes, figure 4a, problems arise in chromosome pairing and prophase I of meiosis, because the two chromosomes are not homologous throughout their length. The homologous regions will pair and undergo synapsis, which often requires that one or both chromosomes loop and twist so that these regions are able to line up, figure 4b. The appearance of this characteristic loop structure during meiosis is one way to detect duplications. Duplications may have major effects on the phenotype. In Drosophila melanogaster, for example, a bar mutant has a reduced number of facets in the eye, making the eye smaller and bar-shaped instead of oval, figure 5. The bar mutant results from a small duplication on the X chromosome, which is inherited as an incompletely dominant X-linked trait. Heterozygous female flies have somewhat smaller eyes. The number of facets is reduced. See figure 5, whereas in homozygous female and hemizygous male flies, the number of facets is greatly reduced. See figure 5c. Occasionally, a fly carries three copies of the bar duplication on its X chromosome. In such mutants, which are termed double bar, the number of facets is extremely reduced. See figure 5d. Bar arises from unequal crossing over, a duplication generating process. Figure 6. Institute for Advanced Studies, Job 83. How does a chromosome duplication alter the phenotype? After all, gene sequences are not altered by duplications, and no genetic information is missing. The only change is the presence of additional copies of normal sequences. The answer to this question is not well understood, but the effects are most likely due to imbalances in the amounts of gene products, abnormal gene dosage. The amount of a particular protein synthesized by a cell is often directly related to the number of copies of its corresponding gene. An individual with three functional copies of a gene often produces 1.5 times as much of the protein encoded by that gene as that produced by an individual with two copies. Because developmental processes often require the interaction of many proteins, they may critically depend on the relative amounts of the proteins. If the amount of one protein increases while the amounts of others remain constant, problems can result. Figure 7. Although duplications can have severe consequences when the precise balance of a gene product is critical to cell function, duplications have arisen frequently throughout the evolution of many eukaryotic organisms and are a source of new genes that may provide novel functions. Human phenotypes associated with some duplications are summarized in Table 1. Deletions. A second type of chromosome rearrangement is a deletion, the loss of a chromosome segment. See figure 3b. A chromosome with segments A, B, C, D, F, G that undergoes a deletion of segment EF would generate the mutated chromosome A, B, C, D, G. A large deletion can be easily detected because the chromosome is noticeably shortened. In individuals heterozygous for deletions, the normal chromosome must loop out during the pairing of homologs and prophase I of meiosis to allow the homologous regions of the two chromosomes to align and undergo synapsis. This looping out generates a structure that looks very much like that seen in individuals heterozygous for duplications. The phenotypic consequences of a deletion depend on which genes are located in the deleted region. If the deletion includes the centromere, the chromosome will not segregate in meiosis or mitosis and will usually be lost. Many deletions are lethal in the homozygous state because all copies of any essential genes located in the deleted region are missing. Even individuals heterozygous for a deletion may have multiple defects for three reasons. First, the heterozygous condition may produce imbalances in the amounts of gene products, similar to the imbalances produced by extra gene copies. Second, deletions may allow recessive mutations on the undeleted chromosome to be expressed because there is no wild type allele to mask their expression. This phenomenon is referred to as pseudodominance. The appearance of pseudodominance in otherwise recessive alleles is an indication that a deletion is present on one of the chromosomes. Third, some genes must be present in two copies for normal function. Such a gene is said to be haploid sufficient. Loss of function mutations in haploid sufficient genes are dominant. Notch is a series of X-linked mutations in Drosophila that often result from chromosome deletions. Notch deletions behave as dominant mutations. When heterozygous for the notch deletion, a fly has wings that are notched at the tips and along the edges. The notch locus is therefore haploid sufficient. A single copy of the gene is not sufficient to produce a wild type phenotype. Females that are homozygous for a notch deletion or males that are hemizygous die early in embryonic development. The notch gene codes for a receptor that normally transmits signals received from outside the cell to the cell's interior and is important in fly development. The deletion acts as a recessive lethal because loss of all copies of the notch gene prevents normal development. In humans, a deletion on the short arm of chromosome 5 is responsible for Creutzfeldt-Jakob syndrome. The name French for cry of the cat derives from the peculiar cat-like cry of infants with this syndrome. A child who is heterozygous for this deletion has a small head, widely spaced eyes, a round face, and mental retardation. Deletion of part of the short arm of chromosome 4 results in another human disorder, Wolff-Hirschhorn syndrome, which is characterized by seizures and by severe mental and growth retardation. A third type of chromosome rearrangement is a chromosome inversion, in which a chromosome segment is inverted, turned 180 degrees. See figure 9.5c. If a chromosome originally had segments A, B, C, D, E, F, G, then chromosome A, B, C represents an inversion that includes segments F. For an inversion to take place, the chromosome must break in two places. Inversions that do not include the centromere, such as A, B, C, are termed paracentric inversions, pair meaning next up, whereas inversions that include the centromere, such as A, B, C, G, are termed paracentric inversions, pair meaning around. Individuals with inversions have neither lost nor gained any genetic material, just the gene order has been altered. Nevertheless, these mutations often have pronounced phenotypic effects. An inversion may break a gene into two parts, with one part moving to a new location and destroying the function of that gene. Even when the chromosome breaks or between genes, phenotypic effects may arise from the inverted gene order in an inversion. Many genes are regulated in a position-dependent manner. If their positions are altered by an inversion, they may be expressed at inappropriate times or in inappropriate tissues. This outcome is referred to as a position effect. Institute for Advanced Studies, Job 85. When an individual is homozygous for a particular inversion, no special problems arise in meiosis, and the two homologous chromosomes can pair and separate normally. When an individual is heterozygous for an inversion, however, the gene order of the two homologs differs, and the homologous sequences can align and pair only if the two chromosomes form an inversion loop. Figure 8. The presence of an inversion loop in meiosis indicates that an inversion is present. Individuals heterozygous for inversions also exhibit reduced recombination among genes located in the inverted region. The frequency of crossing over within the inversion is not actually diminished, but when crossing over does take place, the result is a tendency to produce gametes that are not viable and thus no recombinant progeny are observed. Let's see why this occurs. Figure 9 illustrates the results of crossing over within a paracentric inversion. The individual is heterozygous for an inversion. See figure 9a with one wild type, unmutated chromosome A, B, C, D, F, G, and one inverted chromosome A, B, D, C, F, G. In prophase meiosis, an inversion loop forms, allowing the homologous sequences to pair up. See figure 9b. If a single crossover takes place in the inverted region between segments C and D in figure 9, an unusual structure results. See figure 9c. The two outer chromatids, which did not participate in crossing over, contain original, non-recombinant gene sequences. The two inner chromatids, which did cross over, are highly abnormal. Each has two copies of some genes and no copies of others. Furthermore, one of the four chromatids now has two centromeres and is said to be dicentric. The other lacks a centromere and is ascentric. In anaphase meiosis, the centromeres are pulled toward opposite poles and the two homologous chromosomes separate. This stretches the dicentric chromatid across the center of the nucleus, forming a structure called a dicentric bridge. See figure 9d. Eventually, the dicentric bridge breaks as the two centromeres are pulled farther apart. The acentric fragment has no centromere. Spindle fibers do not attach to it, and so this fragment is not segregated into a nucleus in meiosis and is usually lost. In the second division of meiosis, the chromatids separate and four gametes are produced. See figure 9e. Two of the gametes contain the original, non-recombinant chromosomes A, B, C, D, F, G, and A, B, E, D, C, F, G. The other two gametes contain recombinant chromosomes that are missing some genes. These gametes will not produce viable offspring. Thus, no recombinant progeny result when crossing over takes place within a paracentric inversion. Recombination is also reduced within a paracentric inversion. Figure 10. No dicentric bridges or eccentric fragments are produced, but the recombinant chromosomes have too many copies of some genes and no copies of others, so gametes that receive the recombinant chromosomes cannot produce viable progeny. Figures 9 and 10 illustrate the results of single crossovers within inversions. Double crossovers, in which both crossovers are on the same two-strand, double crossovers result in functional, recombinant chromosomes. Thus, even though the overall rate of recombination is reduced within an inversion, some viable recombinant progeny may still be produced through two-stranded double crossovers. Inversion heterozygotes are common in many organisms, including a number of plants, some species of Drosophila, mosquitoes, and grasshoppers. Inversions may have played an important role in human evolution. Gene banding patterns reveal that several human chromosomes differ from those of chimpanzees by only a paracentric inversion. Figure 11. Translocations. A translocation entails the movement of genetic material between non-homologous chromosomes. See figure 3d or within the same chromosome. Translocation should not be confused with crossing over, in which there is an exchange of genetic material between homologous chromosomes. In non-reciprocal translocations, genetic material moves from one chromosome to another without any reciprocal exchange. Consider the following two non-homologous chromosomes: A, B, C, D, F, G, and M, N, O, P, Q, R, S. If chromosome segment DF moves from the first chromosome to the second without any transfer of segments from the second chromosome to the first, a non-reciprocal translocation has taken place, producing chromosomes A, B, C, D, G, and M, N, O occurs. More commonly, there is a two-way exchange of segments between the chromosomes, resulting in a reciprocal translocation. A reciprocal translocation between chromosomes A, B, C, D, F, G, and M, N, O, P, Q, R, S might give rise to chromosomes A, B, C, D, Q, R, G, and M, N, O, P, S. Translocations can affect a phenotype in several ways. First, they may create new linkage relations that affect gene expression. A position effect genes translocated to new locations may come under the control of different regulatory sequences of other genes that affect their expression. An example is found in Burkitt lymphoma. Second, the chromosomal breaks that bring about translocations may take place within a gene and disrupt its function. Molecular geneticists have used these types of effects to map human genes. Neurofibromatosis is a genetic disease characterized by numerous fibrous tumors of the skin and nervous tissue. It results from an autosomal dominant mutation. Linkage studies first placed the locus for neurofibromatosis on chromosome 17. Geneticists later identified two patients with neurofibromatosis who possessed a translocation affecting chromosome 17. These patients were assumed to have developed neurofibromatosis because one of the chromosome breaks that occurred in the translocation disrupted a particular gene that causes neurofibromatosis. DNA from the regions around the breaks was sequenced and eventually led to the identification of the gene responsible for neurofibromatosis. Institute for Advanced Studies, Job 87. Deletions frequently accompany translocations. In a Robertsonian translocation, for example, the long arms of two acrocentric chromosomes become joined to a common centromere through a translocation, generating a metacentric chromosome with two long arms and another chromosome with two very short arms. Figure 12. The smaller chromosome often fails to segregate, leading to an overall reduction in chromosome number. As we will see, Robertsonian translocations are the cause of some cases of Down syndrome. The effects of a translocation on chromosome segregation in meiosis depend on the nature of the translocation. Let us consider what happens in an individual heterozygous for a reciprocal translocation. Suppose that the original chromosome segments were A, B, C, D, F, G, and M, N, O, P, Q, R, S, designated N1 and N2, and a reciprocal translocation takes place, producing chromosomes A, B, C, D, Q, R, S, and M, N, O, P, F, G, designated T1 and T2. An individual heterozygous for this translocation would possess one normal copy of each chromosome and one translocated copy. Figure 13a. Each of these chromosomes contain segments that are homologous to two other chromosomes. When the homologous sequences pair in prophase meiosis, cross-like configurations consisting of all four chromosomes. Figure 13b form. Notice that N1 and T1 have homologous centromeres in both chromosomes. The centromeres between segments B and C. Similarly, N2 and T2 have homologous centromeres between segments N and O. Normally, homologous centromeres separate and move toward opposite poles in anaphase meiosis. With a reciprocal translocation, the chromosomes may segregate in three different ways. In alternate segregation, figure 13c, N1 and N2 move toward one pole and T1 and T2 move toward the opposite pole. In adjacent one segregation, N1 and T2 move toward one pole and T1 and N2 move toward the other pole. In both alternate and adjacent one segregation, homologous centromeres segregate toward opposite poles. Adjacent two segregation, in which N1 and T1 move toward one pole and T2 and N2 move toward the other, is rare. The products of the three segregation patterns are illustrated in figure 13d. As you can see, the gametes produced by alternate segregation possess one complete set of the chromosome segments. These gametes are therefore functional and can produce viable progeny. In contrast, gametes produced by adjacent one and adjacent two segregation are not viable because some chromosome segments are present in two copies, whereas others are missing. Adjacent two segregation is rare, and so most gametes are produced by alternate and adjacent segregation. Therefore, approximately half of the gametes from an individual heterozygous for a reciprocal translocation are expected to be functional. Translocations can play an important role in the evolution of karyotypes. Chimpanzees, gorillas, and orangutans all have 48 chromosomes, whereas humans have 46. Human chromosome 2 is a large, metacentric chromosome with G-banding patterns that match those found on two different acrocentric chromosomes of the X. Figure 14. Apparently, a Robertsonian translocation took place in a human ancestor, creating a large metacentric chromosome from the two long arms of the ancestral acrocentric chromosomes and a small chromosome consisting of the two short arms. The small chromosome was subsequently lost, leading to the reduced human chromosome number. Fragile sites. Chromosomes of cells grown in culture sometimes develop constrictions or gaps at particular locations called fragile sites. Figure 15 because they are prone to breakage under certain conditions. A number of fragile sites have been identified on human chromosomes. One of the most intensively studied is a fragile site on the human X chromosome that is associated with mental retardation, the Fragile X syndrome. Exhibiting X-linked inheritance and arising with a frequency of about 1 in 12-50 male births, Fragile X syndrome has been shown to result from an increase in the number of repeats of a CGG nucleotide. However, other common fragile sites do not consist of nucleotide repeats, and their nature is still incompletely understood. Aneuploidy. In addition to chromosome rearrangements, chromosome mutations also include changes in the number of chromosomes. Variations in chromosome number can be classified into two basic types: changes in the number of individual chromosomes, aneuploidy, and changes in the number of chromosome sets, polyploidy. Aneuploidy can arise in several ways. First, a chromosome may be lost in the course of mitosis or meiosis if, for example, its centromere is deleted. Loss of the centromere prevents the spindle fibers from attaching, so the chromosome fails to move to the spindle pole and does not become incorporated into a nucleus after cell division. Second, the small chromosome generated by Robertsonian translocation may be lost in mitosis or meiosis. Third, aneuploids may arise through non-disjunction, the failure of homologous chromosomes or sister chromatids to separate in meiosis or mitosis. Non-disjunction leads to some gametes or cells that contain an extra chromosome and others that are missing a chromosome. Figure 15. Institute for Advanced Studies, Job 89. Types of aneuploidy. We will consider four types of relatively common aneuploid conditions in diploid individuals: melisome, monosomy, trisomy, and tetrasomy. 1. Melisome is the loss of both members of a homologous pair of chromosomes. It is represented as 2 and 2, where n refers to the haploid number of chromosomes. Thus, among humans, who normally possess 2n equals 46 chromosomes, a nullosomic person has 44 chromosomes. 2. Monosomy is the loss of a single chromosome, represented as 2 and 1. A monosomic person has 45 chromosomes. 3. Trisomy is the gain of a single chromosome, represented as 2n plus 1. A trisomic person has 47 chromosomes. The gain of a chromosome means that there are 3 homologous copies of one chromosome. 4. Tetrasomy is the gain of 2 homologous chromosomes, represented as 2n plus 2. A tetrasomic person has 48 chromosomes. Tetrasomy is not the gain of any 2 extra chromosomes, but rather the gain of 2 homologous chromosomes, so there will be 4 homologous copies of a particular chromosome. More than one aneuploid mutation may occur in the same individual. An individual that has an extra copy of 2 different non-homologous chromosomes is referred to as being double trisomic and represented as 2n plus 1 plus 1. Similarly, a double monosomic has 2 fewer non-homologous chromosomes, 2n 1 to 1, and a double tetrasomic has 2 extra pairs of homologous chromosomes, 2n plus 2 plus 2. Effects of aneuploid. Effects of aneuploidy. One of the first aneuploids to be recognized was a fruit fly with a single X chromosome and no Y chromosome, which was discovered by Calvin Bridges in 1913. Another early study of aneuploidy focused on mutants in the Ginseng weed, Datura stramonium. Francis Blakesley began breeding this plant in 1913, and he observed that crosses with several Ginseng mutants produced unusual ratios of progeny. For example, the globe mutant having a seed case globular in shape was dominant but was inherited primarily from the female parent. When globe plants were self-fertilized, only 25% of the progeny had the globe phenotype, an unusual ratio for a dominant trait. Blakesley isolated 12 different mutants that also exhibited peculiar patterns of inheritance. Eventually, John Bellin demonstrated that these 12 mutants are in fact trisomics. Datura stramonium has 12 pairs of chromosomes, 2n equals 24, and each of the 12 mutants is trisomic for a different chromosome pair. The aneuploid nature of the mutants explained the unusual ratios that Blakesley had observed in the progeny. Many of the extra chromosomes in the trisomics were lost in meiosis, so fewer than 50% of the gametes carried the extra chromosome, and the proportion of trisomics in the progeny was low. Furthermore, the pollen containing an extra chromosome was not as successful in fertilization, and trisomic zygotes were less viable. Aneuploidy usually alters the phenotype drastically. In most animals and many plants, aneuploid mutations are lethal. Because aneuploidy affects the number of gene copies but not their nucleotide sequences, the effects of aneuploidy are most likely due to abnormal gene dosage. Aneuploidy alters the dosage for some, but not all, genes, disrupting the relative concentrations of gene products and often interfering with normal development. A major exception to the relation between gene number and protein dosage pertains to genes on the mammalian X chromosome. In mammals, X chromosome inactivation ensures that males who have a single X chromosome and females who have 2 X chromosomes receive the same functional dosage for X-linked genes. Extra X chromosomes in mammals are inactivated, so we might expect that aneuploidy of the sex chromosomes would be less detrimental in these animals. Indeed, this is the case for mice and humans, for whom aneuploids of the sex chromosomes are the most common form of aneuploidy seen in living organisms. Y chromosome aneuploids are probably common because there is so little information on the Y chromosome. Aneuploidy in humans. Aneuploidy in humans usually produces serious developmental problems that lead to spontaneous abortion and miscarriage. In fact, as many as 50% of all spontaneously aborted fetuses carry chromosome defects, and a third or more of all conceptions spontaneously abort, usually so early in development that the mother is not even aware of her pregnancy. Only about 2% of all fetuses with a chromosome defect survive to birth. Sex chromosome aneuploids. The most common aneuploidy seen in living humans has to do with the sex chromosomes. As is true of all mammals, aneuploidy of the human sex chromosomes is better tolerated than aneuploidy of autosomal chromosomes. Turner syndrome and Klinefelter syndrome both result from aneuploidy of the sex chromosomes. Autosomal aneuploids. Autosomal aneuploids resulting in live births are less common than sex chromosome aneuploids in humans, probably because there is no mechanism of dosage compensation for autosomal chromosomes. Most autosomal aneuploids are spontaneously aborted, with the exception of aneuploids of some of the small autosomes. Because these chromosomes are small and carry fewer genes, the presence of extra copies is less detrimental. For example, the most common autosomal aneuploid in humans is trisomy 21, also called Down syndrome. The number of genes on different human chromosomes is not precisely known at the present time, but DNA sequence data indicate that chromosome 21 has fewer genes than any other autosome, with perhaps less than 300 genes of a total of 30,000 to 35,000 for the entire genome. The incidence of Down syndrome in the United States is about 1 in 700 human births, although the incidence is higher among children born to older mothers. People with Down syndrome show variable degrees of mental retardation, with an average IQ of about 50 compared with an average IQ of 100 in the general population. Many people with Down syndrome also have characteristic facial features, some retardation of growth and development, and an increased incidence of heart defects, leukemia, and other abnormalities. Approximately 92% of those who have Down syndrome have 3 full copies of chromosome 21, and therefore a total of 47 chromosomes, a condition termed primary Down syndrome. Primary Down syndrome usually arises from random non-disjunction and egg formation. About 75% of the non-disjunction events that cause Down syndrome are maternal in origin, and most arise in meiosis. Most children with Down syndrome are born to normal parents, and the failure of the chromosomes to divide has little hereditary tendency. A couple who has conceived one child with primary Down syndrome has only a slightly higher risk of conceiving a second child with Down syndrome compared with other couples of similar age who have not had any Down syndrome children. Similarly, a couple who has conceived one child with primary Down syndrome has only a slightly higher risk of conceiving a second child with Down syndrome compared with other couples of similar age who have not had any Down syndrome children. Similarly, the couple's relatives are not more likely to have a child with primary Down syndrome. Most cases of primary Down syndrome arise from maternal non-disjunction, and the frequency of this occurrence correlates with maternal age. Although the underlying cause of the association between maternal age and non-disjunction remains obscure, recent studies have indicated a strong correlation between non-disjunction and aberrant meiotic recombination. Most chromosomes that fail to separate in meiosis do not show any evidence of having recombined with one another. Conversely, chromosomes that appear to have failed to separate in meiosis often show evidence of recombination in regions that do not normally recombine, most notably near the centromere. Although aberrant recombination appears to play a role in non-disjunction, the maternal age effect is more complex. In female mammals, prophase begins in oligonia during fetal development, and recombination is completed prior to birth. Meiosis then arrests in diploidy, and the primary oocytes remain suspended until just before ovulation. As each primary oocyte is ovulated, meiosis resonates and the first division is completed, producing a secondary oocyte. At this point, meiosis is suspended again, and remains so until the secondary oocyte is penetrated by a sperm. The second meiotic division takes place immediately before the nuclei of egg and sperm unite to form a zygote. An explanation of the maternal age effect must take into account the apparent recombination that occurs prenatally and the long suspension in prophase I1 theory is that the best oocytes are ovulated first, leaving those oocytes that had apparent recombination to be used later in life. However, evidence indicates that the frequency of apparent recombination is similar between oocytes that are ovulated in young women and those ovulated in older women. Another possible explanation is that aging of the cellular components needed for meiosis results in non-disjunction of chromosomes that are at risk because they have failed to recombine or had some recombination defect. In younger oocytes, these chromosomes can still be segregated from one another, but in older oocytes, they are sensitive to other perturbations in the meiotic machinery. In contrast, sperm are produced continually after puberty, with no long suspension of the meiotic divisions. This fundamental difference between the meiotic process in females and males may explain why most chromosome aneuploidy in humans is maternal in origin. About 4% of people with Down syndrome have 46 chromosomes, but an extra copy of part of chromosome 21 is attached to another chromosome through a translocation, figure 17. This condition is termed familial Down syndrome because it has a tendency to run in families. The phenotypic characteristics of familial Down syndrome are the same as those for primary Down syndrome. Familial Down syndrome arises in offspring whose parents are carriers of chromosomes that have undergone a Robertsonian translocation, most commonly between chromosome 21 and chromosome 14. The long arm of 21 and the short arm of 14 exchange places. This exchange produces a chromosome that includes the long arms of chromosomes 14 and 21, and a very small chromosome that consists of the short arms of chromosomes 21 and 14. The small chromosome is generally lost after several cell divisions. Persons with the translocation, called translocation carriers, do not have Down syndrome. Although they possess only 45 chromosomes, their phenotypes are normal because they have two copies of the long arms of chromosomes 14 and 21, and apparently the short arms of these chromosomes, which are lost, carry no essential genetic information. Although translocation carriers are completely healthy, they have an increased chance of producing children with Down syndrome. When a translocation carrier produces gametes, the translocation chromosome may segregate in three different ways. First, it may separate from the normal chromosomes 14 and 21 in anaphase I meiosis. First, it may separate from the normal chromosomes 14 and 21 in anaphase I meiosis. In this type of Institute for Advanced Studies, Jodhpur 91. Segregation, half of the gametes will have the translocation chromosome and no other copies of chromosomes 21 and 14. The fusion of such a gamete with a normal gamete will give rise to a translocation carrier. The other half of the gametes produced by this first type of segregation will be normal, each with a single copy of chromosomes 21 and 14, and will result in normal offspring. Alternatively, the translocation chromosome may separate from chromosome 14 and pass into the same cell with the normal chromosome 21. This type of segregation produces all abnormal gametes. Half will have two functional copies of chromosome 21, one normal and one attached to chromosome 14, and the other half will lack chromosome 21. The gametes with the two functional copies of chromosome 21 will produce children with familial Down syndrome. The gametes lacking chromosome 21 will result in zygotes with monosomy 21 and will be spontaneously aborted. In the third type of segregation, the translocation chromosome and the normal copy of chromosome 14 segregate together, and the normal chromosome 21 segregates by itself. This pattern is presumably rare, because the two centromeres are both derived from chromosome 14 separately from each other. In any case, all the gametes produced by this process are abnormal. Half result in monosomy 14 and the other half result in trisomy 14. All are spontaneously aborted. Thus, only 3 of the 6 types of gametes that can be produced by a translocation carrier will result in the birth of a baby, and theoretically, these gametes should arise with equal frequency. 1/3 of the offspring of a translocation carrier should be translocation carriers like their parent. 1/3 should have familial Down syndrome, and 1/3 should be normal. In reality, however, fewer than 1/3 of the children born to translocation carriers have Down syndrome, which suggests that some of the embryos with Down syndrome are spontaneously aborted. Few autosomal aneuploids besides trisomy 21 result in human live births. Trisomy 18, also known as Edwards syndrome, arises with a frequency of approximately 1 in 8,000 live births. Babies with Edwards syndrome are severely retarded and have low set ears, a short neck, deformed feet, clenched fingers, heart problems, and other disabilities. Few live for more than a year after birth. Trisomy 13 has a frequency of about 1 in 15,000 live births and produces features that are collectively known as Hot Tau syndrome. Characteristics of this condition include severe mental retardation, a small head, sloping forehead, small eyes, cleft lip and palate, extra fingers and toes, and numerous other problems. About half of children with trisomy 13 die within the first month of life, and 95% die by the age of 3. Rare still is trisomy 8, which arises with a frequency of about 1 in 25,000 to 50,000 live births. This aneuploid is characterized by mental retardation, contracted fingers and toes, low malformed ears, and a prominent forehead. Many who have this condition have normal life expectancy. Uniparental disomy. Normally, the two chromosomes of a homologous pair are inherited from different parents, one from the father and one from the mother. The development of molecular techniques that facilitate the identification of specific DNA sequences has made it possible to determine the parental origins of chromosomes. Surprisingly, sometimes both chromosomes are inherited from the same parent, a condition termed uniparental disomy. Uniparental disomy violates the rule that children affected with a recessive disorder appear only in families where both parents are carriers. For example, cystic fibrosis is an autosomal recessive disease. Typically, both parents of an affected child are heterozygous for the cystic fibrosis mutation on chromosome 7. However, a small proportion of people with cystic fibrosis have only a single parent who is heterozygous for the cystic fibrosis gene. How can this be? These people must have inherited from the heterozygous parent two copies of the chromosome 7 that carries the defective cystic fibrosis allele and no copy of the normal allele from the other parent. Uniparental disomy has also been observed in Prader-Willi syndrome, a rare condition that arises when a paternal copy of a gene on chromosome 15 is missing. Although most cases of Prader-Willi syndrome result from a chromosome deletion that removes the paternal copy of the gene, from 20% to 30% arise when both copies of chromosome 15 are inherited from the mother and no copy is inherited from the father. Many cases of uniparental disomy probably originate as a trisomy. Although most autosomal trisomies are lethal, a trisomic embryo can survive if one of the 3 chromosomes is lost early in development. If, just by chance, the 2 remaining chromosomes are both from the same parent, uniparental disomy results. Mosaicism. Non-disjunction in a meiotic division may generate patches of cells in which every cell has a chromosome abnormality and other patches in which every cell has a normal karyotype. This type of non-disjunction leads to regions of tissue with different chromosome constitutions, a condition known as mosaicism. Growing evidence suggests that mosaicism is relatively common. Only about 50% of those diagnosed with Turner syndrome have the 45X karyotype presence of a single X chromosome in all their cells. Most others are mosaics, possessing some 45X cells and some normal 46XX cells. A few may even be mosaics for 2 or more types of abnormal karyotypes. The 45X46XX mosaic usually arises when an X chromosome is lost soon after fertilization in an XX embryo. Fruit flies that are XXXO mosaics. O designates the absence of a homologous chromosome. XO means the cell has a single X chromosome and no Y chromosome. Develop a mixture of male and female traits. Because the presence of 2 X chromosomes in fruit flies produces female traits and the presence of a single X chromosome produces male traits. Figure 18. Sex determination in fruit flies occurs independently in each cell during development. Those cells that are XX express female traits. Those that are XY express male traits. Such sexual mosaics are called gynandromorphs. Normally, X-linked recessive genes are masked in heterozygous females, but in XXXO mosaics, any X-linked recessive genes present in the cells with a single X chromosome will be expressed. Uniparental disomy. Normally, the 2 chromosomes of a homologous pair are inherited from different parents, one from the father and one from the mother. The development of molecular techniques that facilitate the identification of specific DNA sequences has made it possible to determine the parental origins of chromosomes. Surprisingly, sometimes both chromosomes are inherited from the same parent, a condition termed uniparental disomy. Uniparental disomy violates the rule that children affected with a recessive disorder appear only in families where both parents are carriers. For example, cystic fibrosis is an autosomal recessive disease. Typically, both parents of an affected child are heterozygous for the cystic fibrosis mutation on chromosome 7. However, a small proportion of people with cystic fibrosis have only a single parent who is heterozygous for the cystic fibrosis gene. How can this be? These people must have inherited from the heterozygous parent 2 copies of the chromosome 7 that carries the defective cystic fibrosis allele and no copy of the normal allele from the other parent. Uniparental disomy has also been observed in Prader-Willi syndrome, a rare condition that arises when a paternal copy of a gene on chromosome 15 is missing. Although most cases of Prader-Willi syndrome result from a chromosome deletion that removes the paternal copy of the gene, from 20% to 30% arise when both copies of chromosome 15 are inherited from the mother and no copy is inherited from the father. Many cases of uniparental disomy probably originate as a trisomy. Although most autosomal trisomies are lethal, a trisomic embryo can survive if one of the 3 chromosomes is lost early in development. If, just by chance, the 2 remaining chromosomes are both from the same parent, uniparental disomy results. Mosaicism. Non-disjunction in a meiotic division may generate patches of cells in which every cell has a chromosome abnormality and other patches in which every cell has a normal karyotype. This type of non-disjunction leads to regions of tissue with different chromosome constitutions, a condition known as mosaicism. Growing evidence suggests that mosaicism is relatively common. Non-disjunction in a meiotic division may generate patches of cells in which every cell has a chromosome abnormality and other patches in which every cell has a normal karyotype. This type of non-disjunction leads to regions of tissue with different chromosome constitutions, a condition known as mosaicism. Non-disjunction in a meiotic division may generate patches of cells in which every cell has a chromosome abnormality and other patches in which every cell has a normal karyotype. This type of non-disjunction leads to regions of tissue with different chromosome constitutions, a condition known as mosaicism. Growing evidence suggests that mosaicism is relatively common. Only about 50% of those diagnosed with Turner syndrome have a 45X karyotype presence of a single X chromosome in all their cells. Most others are mosaics, possessing some 45X cells and some normal 46XX cells. A few may even be mosaics for 2 or more types of abnormal karyotypes. The 45X46XX mosaic usually arises when an X chromosome is lost soon after fertilization in an XX embryo. Fruit flies that are XXXO mosaics O designates the absence of a homologous chromosome. XO means the cell has a single X chromosome and no Y chromosome. Develop a mixture of male and female traits. Because the presence of 2 X chromosomes in fruit flies produces female traits and the presence of a single X chromosome produces male traits. Figure 18. Sex determination in fruit flies occurs independently in each cell during development. Those cells that are XX express female traits. Those that are XY express male traits. Such sexual mosaics are called gyneandromorphs. Normally, X-linked recessive genes are masked in heterozygous females, but in XXXO mosaics, any X-linked recessive genes present in the cells with a single X chromosome will be expressed. Polyploidy. Most eukaryotic organisms are diploid 2N for most of their life cycles, possessing 2 sets of chromosomes. Occasionally, whole sets of chromosomes fail to separate in meiosis or mitosis, leading to polyploidy, the presence of more than 2 genomic sets of chromosomes. Polyploids include triploids, 3N tetraploids, 4N pentaploids, 5N, and even higher numbers of chromosome sets. Polyploidy is common in plants and is a major mechanism by which new plant species have evolved. Approximately 40% of all flowering plant species and from 70% to 80% of grasses are polyploids. They include a number of agriculturally important plants, such as wheat, oats, cotton, potatoes, and sugarcane. Polyploidy is less common in animals, but is found in some invertebrates, fishes, salamanders, frogs, and lizards. No naturally occurring, viable polyploids are known in birds, but at least one polyploid mammal, a rat from Argentina, has been reported. We will consider 2 major types of polyploidy: autopolyploidy, in which all chromosome sets are from a single species, and allopolyploidy, in which chromosome sets are from 2 or more species. Autopolyploidy. Autopolyploidy results when accidents of meiosis or mitosis produce extra sets of chromosomes, all derived from a single species. Non-disjunction of all chromosomes in mitosis in an early 2N embryo, for example, doubles the chromosome number and produces an autotetraploid 4N figure 19a. An autotriploid may arise when non-disjunction in meiosis produces a diploid gamete that then fuses with a normal haploid gamete to produce a triploid zygote figure 19b. Alternatively, triploids may arise from a cross between an autotetraploid that produces 2N gametes and a diploid that produces 1N gametes. Because all the chromosome sets in autopolyploids are from the same species, they are homologous in attempt to align in prophase of meiosis, which usually results in sterility. Because all the chromosome sets in autopolyploids are from the same species, they are homologous in attempt to align in prophase of meiosis, which usually results in sterility. Consider meiosis in an autotriploid figure 20. In meiosis in a diploid cell, 2 chromosome homologs pair in a line, but in autotriploids, 3 homologs are present. One of the 3 homologs may fail to align with the other 2, and this unaligned chromosome will segregate randomly. See figure 20a. Which gamete gets the extra chromosome will be determined by chance and will differ for each homologous group of chromosomes. The resulting gametes will have 2 copies of some chromosomes and 1 copy of others. Even if all 3 chromosomes do align, 2 chromosomes must segregate to 1 gamete and 1 chromosome to the other. See figure 20b. Occasionally, the presence of a third chromosome interferes with normal alignment, and all 3 chromosomes segregate to the same gamete. See figure 20c. No matter how the 3 homologous chromosomes align, their random segregation will create unbalanced gametes with various numbers of chromosomes. A gamete produced by meiosis in such an autotriploid might receive, say, 2 copies of chromosome 1, 1 copy of chromosome 2, 3 copies of chromosome 3, and no copies of chromosome 4. When the unbalanced gamete fuses with a normal gamete or with another unbalanced gamete, the resulting zygote has different numbers of the 4 types of chromosomes. This difference in number creates unbalanced gene dosage. Institute for Advanced Studies, Job 93. In the zygote, which is often lethal. For this reason, triploids do not usually produce viable offspring. In even-numbered autopolyploids, such as autotetraploids, it is theoretically possible for the homologous chromosomes to form pairs and divide equally. However, this event rarely happens, so these types of autotetraploids also produce unbalanced gametes. The sterility that usually accompanies autopolyploidy has been exploited in agriculture. Wild diploid bananas 2N equals 22, for example, produce seeds that are hard and inedible, but triploid bananas 3N equals 33 are sterile and produce no seeds. They are the bananas sold commercially. Similarly, seedless triploid watermelons have been created and are now widely sold. Allopolyploidy. Allopolyploidy arises from hybridization between 2 species. The resulting polyploid carries chromosomes sets derived from 2 or more species. Figure 21 shows how allopolydy can arise from 2 species that are sufficiently related that hybridization occurs between them. Species I AABBCC 2N equals 6 produces haploid gametes with chromosomes ABC, and species 2 GGHHI 2N equals 6 produces gametes with chromosomes GHI. If gametes from species I and 2 fuse, a hybrid with 6 chromosomes ABCGHI is created. The hybrid has the same chromosome number as that of both diploid species, so the hybrid is considered diploid. However, because the hybrid chromosomes are not homologous, they will not pair and segregate properly in meiosis, so this hybrid is functionally haploid and sterile. The sterile hybrid is unable to produce viable gametes through meiosis, but it may be able to perpetuate itself through mitosis asexual reproduction. On rare occasions, non-disjunction takes place in a meiotic division, which leads to a doubling of chromosome number in an allotetraploid with chromosomes ABCGHHT. This tetraploid is functionally diploid. Every chromosome has one and only one homologous partner, which is exactly what meiosis requires for proper segregation. The allopolyploid can now undergo normal meiosis to produce balanced gametes having 6 chromosomes. George Karpachenko created polyploids experimentally in the 1920s. Today, as well as in the early 20th century, cabbage Brassica oleracea 2N equals 18 and radishes Raphanus sativa 2N equals 18 are agriculturally important plants, but only the leaves of the cabbage and the roots of the radish are normally consumed. Karpachenko wanted to produce a plant that had cabbage leaves and radish roots so that no part of the plant would go to waste. Because both cabbage and radish possess 18 chromosomes, Karpachenko was able to successfully cross them, producing a hybrid with 2N equals 18, but unfortunately, the hybrid was sterile. After several crosses, Karpachenko noticed that one of his hybrid plants produced a few seeds. When planted, these seeds grew into plants that were viable and fertile. Analysis of their chromosomes revealed that the plants were allotetraploids with 2N equals 36 chromosomes. To Karpachenko's great disappointment, however, the new plants possess the roots of a cabbage and the leaves of a radish. The significance of polyploidy. In many organisms, cell volume is correlated with nuclear volume, which, in turn, is determined by genome size. Thus, the increase in chromosome number in polyploidy is often associated with an increase in cell size, and many polyploids are physically larger than diploids. Breeders have used this effect to produce plants with larger leaves, flowers, fruits, and seeds. The hexaploid 6N equals 42 genome of wheat probably contains chromosomes derived from 3 different wild species. Figure 22. Many other cultivated plants also are polyploid. Table 2. Polyploidy is less common in animals than in plants for several reasons. As discussed, allopolyploids require hybridization between different species, which occurs less frequently in animals than in plants. Animal behavior often prevents interbreeding, and the complexity of animal development causes most interspecific hybrids to be non-viable. Many of the polyploid animals that do arise are in groups that reproduce through parthenogenesis, a type of reproduction in which individuals develop from unfertilized eggs. Thus, asexual reproduction may facilitate the development of polyploids, perhaps because the perpetuation of hybrids through asexual reproduction provides greater opportunities for non-disjunction. Only a few human polyploid babies have been reported, and most died within a few days of birth. Polyploidy, usually triploidy, is seen in about 10% of all spontaneously aborted human fetuses. Different types of chromosome mutations are summarized in Table 3. Chromosome mutations in cancer. Most tumors contain cells with chromosome mutations. For many years, geneticists argued about whether these chromosome mutations were the cause or the result of cancer. Some types of tumors are consistently associated with specific chromosome mutations. Most tumors contain cells with chromosome mutations. For many years, geneticists argued about whether these chromosome mutations were the cause or the result of cancer. Some types of tumors are consistently associated with specific chromosome mutations, suggesting that in these cases a specific chromosome mutation played a pivotal role in the development of the cancer. However, Institute for Advanced Studies, Job 95. Many cancers are not associated with specific types of chromosome abnormalities, and individual gene mutations are now known to contribute to many types of cancer. Nevertheless, chromosome instability is a general feature of cancer cells, causing them to accumulate chromosome mutations, which then affect individual genes that contribute to the cancer process. Thus, chromosome mutations appear to both cause and be a result of cancer. At least 3 types of chromosome rearrangements, deletions, inversions, and translocations are associated with certain types of cancer. Deletions may result in the loss of one or more genes that normally hold cell division in check. When these so-called tumor suppressor genes are lost, cell division is not regulated and cancer may result. Inversions and translocations contribute to cancer in several ways. First, the chromosomal breakpoints that accompany these mutations may lie within tumor suppressor genes, disrupting their function and leading to cell proliferation. Second, translocations and inversions may bring together sequences from 2 different genes, generating a fused protein that stimulates some aspect of the cancer process. Such fusions are seen in most cases of chronic myeloid leukemia, a fatal form of leukemia affecting bone marrow cells. About 90% of patients with chronic myeloid leukemia have a reciprocal translocation between the long arm of chromosome 22 and the tip of the long arm of chromosome 9, figure 23. This translocation produces a shortened chromosome 22, called the Philadelphia chromosome because it was first discovered in Philadelphia. At the end of a normal chromosome 9 is a potential cancer-causing gene called CABL. As a result of the translocation, part of the CABL gene is fused with the BCR gene from chromosome 22. The protein produced by this BCR-CABL fusion gene is much more active than the protein produced by the normal CABL gene. The fusion protein stimulates increased and regulated cell division and eventually leads to leukemia. A third mechanism by which chromosome rearrangements may produce cancer is by the transfer of a potential cancer-causing gene to a new location, where it is activated by different regulatory sequences. Burkitt lymphoma is a cancer of the B cells, the lymphocytes that produce antibodies. Many people having Burkitt lymphoma possess a reciprocal translocation between chromosome 8 and chromosome 2, 14, or 22, each of which carries genes for immunological proteins, figure 24. This translocation relocates a gene called CMYC from the tip of chromosome 8 to a position in one of the aforementioned chromosomes that is next to a gene for one of the immunoglobulin proteins. At this new location, CMYC comes under the control of regulatory sequences that normally activate the production of immunoglobulins, and CMYC is expressed in B cells. The CMYC protein stimulates the division of the B cells and leads to Burkitt lymphoma. K recombination, homologous and non-homologous recombination, including transposition, site-specific recombination. Introduction. A biological significance of mobile DNA elements. The development of an interest in the ID nature of mobile genetic elements. Barbara McClintock seminar papers describing eukaryotic transposition of what she called controlling elements in Maze during the 1950s. McClintock demonstrated the movement of specific genetic elements to new chromosomal locations affecting the expression of nearby genes and cause chromosome breakages in a developmentally regulated manner. Since McClintock's remarkable discovery, the dynamic nature of mobile genetic elements has been observed in almost every prokaryotic and eukaryotic organisms investigated to date. The genome of any organism must possess 2 key features. First, it must be stable enough to pass accurate information through inheritance, ensuring the survival of progeny. However, the genome must also be dynamic in order to respond to selective environmental pressures. Therefore, any successful biological system must maintain a delicate balance between genome integrity and flexibility. In both prokaryotic and eukaryotic systems, recombination is one of the key mechanisms that regulates genome integrity. Chromosomal breakages and mutations, stemming from problems in DNA replication or environmental stress, can be repaired through recombination pathways. In some cases, organisms contain multiple recombination pathways by which damage can be repaired, underscoring the importance of this process. The mobility of DNA sequence elements is also a driving force in evolutionary patterns. Recombination provides a mechanism by which DNA can be moved, deleted, and amplified to affect these changes. These movements are often tightly regulated, such as those involved in gene rearrangements, DNA amplification and deletion, and genome integration events. In other examples, the movements are rare and only become visible when selective pressures are imposed on large populations. One of the most startling examples of recombination-driven evolution involves the inheritance of antibiotic resistance genes among certain populations of bacteria. The biological consequences of recombination are ubiquitous. In recent years, recombination has become an invaluable tool in the biological laboratory for both genome manipulation and genetic analysis. Understanding mechanisms of recombination is therefore central to any discussion of the functional properties of any particular genome. Moreover, recombination systems offer the opportunity to harness the power of mobile DNA elements for use in genetic therapies and other medical applications. B. Genetic recombination, background and perspective. The modern field of recombination, by most accounts, began in 1964 with Robin Holliday's hypothetical four-stranded DNA structure, Holliday, 1964, figure 1. This structure, later to be named the Holliday junction, consists of 2 DNA duplexes associated by single-stranded crossover and was proposed to explain gene conversions previously observed in fungi. Holliday possessed one advantage over McClintock in her earlier work, knowledge of the structure of DNA. Knowing that DNA was double-stranded, he proposed a mechanism by which 2 chromosomes in close proximity could exchange DNA strands, thereby affecting genomic alterations. He also noted the necessity for homology between the exchanging strands. Historically, the Holliday junction and the necessity for extensive DNA homology have defined classical recombination, but it is important to note that there are many different recombination reaction mechanisms that proceed through a variety of intermediate structures. 2. Recombination systems. A general or homologous recombination. The most familiar recombination systems are those that are involved in general or homologous recombination. This class of recombination events is defined by the exchange of homologous sequences between double-stranded DNA molecules and results in recombinant product molecules that contain genetic information originally present in each of the parental molecules. There is generally no limit to how much DNA can be exchanged in this process, ranging from Institute for Advanced Studies, Jodhpur 97. Tens to thousands of base pairs, so long as homology is maintained between recombining duplexes. The basis of recognition in homologous systems is pairing of the exchanging DNA sequences. Protein components of these systems have other roles in the recombination reactions such as DNA strand juxtaposition, recruitment of cofactors such as ATP or accessory proteins, catalysis of strand cleavage and rejoining reactions, and heteroduplex extension or branch migration. A key intermediate in general recombination pathways is the four-stranded Holliday junction, figure 1. One of the hallmarks of this four-way junction is its ability to undergo branch migration, which can extend the heteroduplex region for thousands of base pairs, figure 2. Branch migration can occur spontaneously by thermal fluctuations, resulting in unidimensional diffusion of the junction's branch point, or can be driven by helicase-dependent ATP hydrolysis. The final step of homologous recombination involves resolution of the four-way junction into 2 distinct duplex molecules. Resolution in homologous systems can occur in a variety of ways, depending on the specificity of resolving enzymes and the particular conformation of the DNA intermediate. The locations of strand cleavages that resolve the junction direct the formation of particular products. The locations of strand cleavages that resolve the junction direct the formation of particular products. Homologous recombination systems are involved in numerous cellular functions, including the repair of genomic damage caused by mismatched base pairs, chromosomal breaks or deletions, meaning type conversion, antigenic variation, DNA replication, and meiosis. B. Homologous recombination in Escherichia coli. The paradigm for general recombination is abstracted from a set of record-dependent homologous recombination systems of E. coli. Although our understanding of other homologous recombination systems in both prokaryotic and eukaryotic organisms is rapidly improving, none of these approach the extent to which a record-dependent pathway has been characterized. At least 25 proteins have been shown to play some role in all types of homologous recombination in E. coli. Many of these proteins have functional homologs in other organisms, though there is often little structural homology. The strand exchange protein RECA plays a central role in nearly all E. coli homologous recombination pathways. The conservation of functional RECA homologs underscores the biological importance of DNA strand exchange proteins. All free living organisms examined to date possess a RECA-like protein. Record-dependent homologous recombination involves at least 4 distinct stages, figure 2. 1. Initiation. Initiation encompasses the processing of DNA at a double-stranded break to generate a single-stranded DNA segment required for strand division of a duplex DNA homolog by RECA ssDNA complex. The DNA processing that occurs during initiation in E. coli is carried out by the recombination-specific helicases REC-BCD and REC, probably in conjunction with some exogenous exonuclease activity, such as that of the protein RECJ. In addition to helicase activity, the REC-BCD complex also has an intrinsic nuclease activity. The specificity of this nuclease activity is modulated dramatically by the presence of an APP DNA sequence element, chi, which functions as a hotspot for homologous recombination. Upon encountering a chi site in the appropriate orientation, REC-BCD switches from a 3'5' to a 5'3" exonuclease activity. This strand polarity switch in the exonuclease activity of REC-BCD leads to the preferential formation of DNA molecules bearing a 3' terminal single-stranded overhang, which are ideal substrates for formation of a RECA ssDNA filament known as the presynaptic filament. 2. Synapsis. This stage involves steps that lead to homologous pairing and strand exchange. In the presence of ATP or non-hydrolyzable ATP analogs, RECA protein binds in a highly cooperative manner to single-stranded DNA to produce a nucleoprotein filament in which the ssDNA is stretched to nearly 1.5 times its original length. Both in vitro and in vivo, binding of RECA to ssDNA is assisted by E. coli single-stranded binding protein (SSB), which facilitates RECA binding through the destabilization of internal DNA secondary structure. Although ATP binding stabilizes the ssDNA specific form of RECA, F hydrolysis is not required either for formation of the presynaptic filament or for homologous pairing or strand exchange. Synapsis occurs between the presynaptic filament, which contains the invading segment of single-stranded DNA, and a double-stranded homologous target sequence. Binding of the presynaptic filament to dsDNA is initially random, but the recast DNA filament carries out a rapid and efficient search for sequence homology by a mechanism not fully understood at present. Upon locating the homologous target sequence, the rapid DNA filament generates what is known as a joint molecule, in which the invading single strand displaces a complementary strand of the duplex to form an ED loop. Current structural and biochemical data overwhelmingly support a model for a joint molecule replica complex that contains 3 DNA strands. This view is in contrast to previously proposed models that invoke a 4-stranded DNA intermediate. Thus it seems